Good evening and welcome to this amazing webinar that has been put together with a lot of care and effort. We're seeking God's blessings on us as we try to unravel some of these very important secrets. It is my proud privilege on behalf of the Believers Church Medical College Hospital to welcome a dear friend, Professor Sita Naik, former professor and head of clinical immunology as SGPGI Lucknow, Professor Abel Lawrence, Professor of Clinical Immunology at SGPGI, Dr. Padmanabha Shenoy, the Medical Director of Care in Kochi, Dr. Vishad Vishwanath, Medical Director of IRIS in Trivandrum, our very own Dr. Vishnu Chandran of our Rheumatology and Immunology Department, and my dear friend and colleague, Professor Debashish Danda, founder of our Rheumatology and Clinical Immunology at CMC Vellore, President-elect of APLAR, former President of IRA. Debashish, thank you so much for putting this together. We requested you because we were finding the whole thing so confusing. We couldn't understand what was going on. And we realized that the secret lay in understanding immunology. It just means that all of you immunologists are at the center of the universe at the moment in trying to understand the future of our very livelihood and living. So therefore, let me hand it over to you so that you'll advise us, teach us, and we will learn from you and then take things forward. Over to Professor Debashish Danda.
Good evening. It's my pleasure to uh, be part of this program, to be associated with Dr. George Chandy, who was our director before at CMC Vellore. In many ways, we are indebted to him for many things, including our department, which was possible in CMC Vellore because of him as the director. As Sir has highlighted, the need for understanding of immunology has now gone beyond the the narrow spectrum of the specialty of immunology. I think everybody needs to know a little bit of immunology, and the COVID has compelled us to go in this direction. So today we have uh, a very prominent faculty. The program goes like this. So I start with a overview of the normal immune system, and followed by which we have Dr. Abel Lawrence from SGPGI Lucknow, who is a professor there, and he's of late have been very well uh, informed in COVID science, and he has been constantly giving interviews in CNN and all the national television channels, and he's very well informed with a lot of data which he has gathered and he has mastered in understanding. So he'll speak on how COVID evades and tricks the immune system. After that, Dr. Vishad Vishnath, he is the director of the IRIS, the Institute of Rheumatology and Immunology Science at Thiruvananthapuram. Dr. Vishad has done, after his MBBS and MD from Trivandrum, he had gone to SGPGI to do his DM, and he will speak on how does COVID cause immune-mediated injury. So it will be somewhat linked to what Dr. Abel Lawrence talks in real life scenario. Following which we have another very prominent rheumatologist from Kerala, Dr. Padmanabha Shenoy. He will speak on the role of immunosuppressants and immunomodulators, and also the safety of using them in COVID. So what are the consensus and what are the controversies in this area? After Dr. Shenoy, we have a very eminent speaker and my teacher, uh, Professor Sita Naik, who was the Dean of SGPGI as well as the head of the Department of Clinical Immunology at SGPGI when I was a student there. And Professor Sita Naik will speak on towards COVID vaccine, the challenges and opportunities. And finally, we have the the host speaker from BCMCH, Dr. Vishnu Chandran, who had done his DM in rheumatology from Amrita Institute at Kochi, and he is going to speak on pediatric COVID. Is it an immunological mimic of Kawasaki's disease, as the children present totally differently than adults with COVID infection. And then we have a period of question and answer, and finally I would request Dr. George Chandy to give the closing remarks. So with this outline, I would like to invite you to my talk. If I can share my slides now, I will start off straight without wasting much time on my talk. Uh, so uh, the this will be a very basic overview of almost everything that happens in the immune system. So it will be a lot of information, and I will try to present as simple as possible, keeping the audience in mind from basic anatomy physiology till the higher specialties across various specialties. So the immune system broadly comprises of these areas: the immune organs, the immune cells, the effector immune molecules, and the functions of the immune organs and the cells and the molecules. The immune organs include the primary lymphoid organs like thymus and the bone marrow, and uh, secondary lymphoid organs like spleen, lymph nodes, and the MALT or the mucosal associated lymphoid tissue, largely in relation to GIT and the respiratory airways. And the thymus, as you know, is just above the heart, a bilobed capsular gland which has got a outer cortex and inner medulla. The functions of thymus mainly is maturation of T cells, enhanced diversity of T cells, T cell selection. So if you see the thymus structure, there is a capsule, and then we have the trabeculae, we have the cortex and the medulla. The cortex are the dividing thymocytes, the mature thymocytes and dead cells, and there are also nurse cells which provide nutrition and nurse those thymocytes. There are also cortical epithelial cells apart from macrophages and interdigitating dendritic cells which may be present both in the cortex and medulla. The medulla has all the blood vessels, the Hassel's corpuscles, medullary epithelial cells, interdigitating cells, the various macrophages, etc. So this is roughly about the thymus. Uh, the T cell selection as I have just mentioned as a function of thymus is based on MHC antigen complex recognition. Basically, the capacity to recognize MHC non-self antigen complex, recognize MHC self antigen complex, and do not recognize MHC antigen complexes. So these are the various areas on which the thymus has to work on, and as we know, if the thymus is not there, it leads on to primary immune deficiency states, and even thymectomized people develop various complications. So T cell maturation usually happens in 8th or 9th week of gestation in humans. There are two terms called positive selection and negative selection. The positive selection permits the survival of only those T cells which whose TCR are capable of recognizing self MHC molecules, and negative selection eliminates T cells that react too strongly with self MHC or with self MHC plus self peptides. So the bone marrow, on the other hand, has structures which microscopically involve stromal cells, and functions include, apart from hematopoiesis, B cell maturation, B cell selection, and putting out mature naive B cells. The B cell development again has an antigen independent and an antigen dependent pathway. The antigen independent pathway is where the stem cells grow into pro B cell and pre B cells, and to immature B cell onwards up to mature recirculating B cells, germinal center B cells, memory B cells, and plasma cells. That is antigen dependent pathway. So this picture probably can say this better. So you have in the bone marrow, we have pro B's, we have stem cells which grows into pro B cells, pre B cells, and immature B cells in an antigen independent manner, whereas when it goes into peripheral compartments, it becomes from naive B cells on exposure to antigens, it become naive activated B cells, germinal center B cells, post germinal center B cells, memory B cells, plasma blasts, and eventually plasma cells, which actually again travel back to the bone marrow. So this is how the antigen independent and antigen dependent compartments of the B cell development is generally described. Uh, the secondary lymphoid organs include spleen, lymph nodes, and the malt, as I mentioned. The lymph nodes grossly are bean-shaped structures, drains major segments of the lymphatic system. The structurally, microscopically, the major cells it has include lymphocytes, macrophages, and dendritic cells. It has also cortex, paracortex, and medulla, and the follicles like primary and secondary follicle. So if you see here, you have got you can see that here are the efferent lymphatics vessels and the efferent lymphatics and arteries and veins. You have the capsule, and then you have the cortical sinus, you have the marginal sinus, and you have the medullary sinus. And we have the primary lymphoid follicles containing mostly B cells. We have secondary lymphoid follicles which contain germinal centers. Here, when the senescent germinal center becomes small and atrophic, and we have paracortical area which contains predominantly T cells. Apart from that, there are medullary cord, macrophages, and plasma cells containing medullary cords, and there is medullary sinus, as I have mentioned to you. So the same picture, efferent and the efferent and the lymphatic vein and lymphatic artery. Here the post capillary venules. This is a cross section of a post capillary venule, and we have also the B lymphocytes, as you can see in the germinal centers, and these are the primary follicles. This is the germinal centers again, and B lymphocytes in the germinal centers. So this is how a lymph node is like grossly. The function of lymph nodes lymph nodes are the first line of response to antigens. The secondary follicles containing germinal center is the site of B cell proliferation differentiation. The specificity is high, and more than 90% of B cells die through apoptosis or a programmed cell death. After antigen stimulation, lymphocyte numbers can go up by 50 times in the efferent lymphatic vessels, and it can also be responsible for lymphadenopathy when the lymph nodes are activated by antigen presentation, etc. This is about spleen, which has got a gastric surface, got hilus, it's got renal surface, and the splenic vein and arteries. And spleen again microscopically compartmentalized into red pulp and white pulp, and there is also a structure called periarteriolar lymphoid sheath, which is the site of antigen presentation. The main functions of spleen include filtering out old RBCs, responding to antigen in circulatory system, producing activated B cells. So this is like the red pulp here and white pulp here, and we have the trabecular artery here and the trabecular vein here, and we have the venous sinus in between. So this is red pulp, and the white pulp is basically the cells. So this is the transverse section of the white pulp. So you can see the red pulp outside colored pink, and then we have the perifollicular zone, then we have the marginal zone, and we have the B cell corona, we have the germinal center, and here we have the periarteriolar lymphoid sheath and got the central arteriole in between, and the same thing is shown in the longitudinal section here. The white pulp or lymphatics usually have small arterioles surrounded by sheaths of lymphocytes known as periarteriolar lymphoid sheath. Is very important structure in white pulp, and it is surrounded by marginal zone, and as I have told, pals is the one which is responsible for antigen presentation, whereas white pulp clears RBCs. There are cords of cells include erythrocyte, macrophages, dendritic cells, few lymphocytes and plasma cells. Also contains venous sinusoids. The malt or mucosal lymphoid tissue is important in the mucosal surfaces like GIT and the respiratory tract, and the mucosal surface of mouth, the respiratory and reproductive tracts are also colonized by lymphocytes and accessory cells, and they respond to ingested inhaled antigens. Uh, there are also tonsils, Peyer's patches, and appendix as a part of the mucosal associated system related structures. So you have the intestinal lumen, we have the inductive site, we have the villi, and we have the M cells here, and we have subsequent lamina propria, and in the submucosa we have the primary follicles, and we have the muscle layer outside. So this is together is the Peyer's patch. So the malt and Peyer's patches actually involve, they have the lymphoid tissue below the epithelium, and their presence of B cells, antigen presented through unique cells or M cells as we have mentioned, and they preferentially respond with IgA antibodies. As you can see here again, you have the M cells, you have the germinal center here, and you have the follicles here, you have the T cells here, and the efferent. And what happens is, if you see the antigen which enters through the M cells, and they stimulate the organized lymphoid follicles which actually produce mature plasma cells producing IgA, which can traverse out and fight invading organisms. So mucosal epithelium has, apart from M cells, there are also intraepithelial lymphocytes. So tonsils are other follicular structures containing lymphocyte, macrophages, mast cells. Germinal centers appear in response to antigen even in tonsils like lymph nodes, and they are protective. They have protective role in upper respiratory infection. Same is appendix is associated with the intestine response to antigen. It has also got role in GI immune response. So there are also movements of lymphocytes within. This is from red marrow, the B cells traverse into the other lymphatic tissues, where through the lymph nodes they mature into plasma cells and they become immunocompetently, or they can, the plasma cells can also go and travel to spleen and tonsils. On the other hand, the T stem cells come from the red marrow to the thymus as we have mentioned, and they become immunocompetent T cells, and they again travel to other lymphoid tissues as well as into the uh, into the red marrow. Now, immune cells is interestingly have origin from the hematopoietic stem cells, which are self-renewing cells. So there are bone marrow. Uh, in the bone marrow, they develop myeloid progenitor and the lymphoid progenitor cells, and the lymphoid progenitor cells in the peripheral tissue can grow into natural killer cells, or in the thymus they can grow into T cell progenitor to eventually to T helper and T cytotoxic cells, and also into B cells from B cell progenitors. Also from lymphoid progenitors there can be, they can develop in the peripheral tissue dendritic cells. On the other hand, we have the myeloid progenitor cells which can grow into myeloid derived dendritic cells, different from the lymphoid derived dendritic cells. The myeloid can also grow into the granulocytic lineage to neutrophils, or into the macrophages through monophage, from monocyte to macrophages with the interaction of dendritic cells, and you can also have the eosinophil lineage. You can have the basophil lineage going into mast cells, and also have megakaryocytic lineage and the erythroid lineage. So you can see that all the immune cells originally come from hematopoietic stem cells, and out of all the cell, T cells, B cells, and T cells are possibly the most important. So also the phagocytes, all the or the granulocytes. The neutrophils comprise of 50 to 70% of water which stays 7 to 10 hours in blood, then migrates into tissue. The first responders they are, and they are motile and phagocytic. The leukocytosis is seen in infection, and extracellular neutrophil extracellular traps are very important pathway or mechanisms in lupus and many autoimmune diseases. The the various monocytes are mononuclear. They circulate in blood for about 8 hours. They mature into macrophages or dendritic cells or into tissue macrophages.
I'll
They
not have it here,
digest or
but
present
yeah,
antigen
I'll also
uh,
go.
and surface receptors for antibodies. Also, they act as through the opsonized antigens. Uh, the dendritic cells can be, as I have told, heterogeneous, can be myeloid or lymphoid origin, and they can be antigen presenting cells for presenting to naive T cells. They can be actively extending or retracting sampling antigens, and they capture antigens in one place and then migrate and present antigen to another place. So almost like with the movement like amoeba, they can move. The induction of inflammation and homing of activated dendritic cells into the lymph nodes is an interesting interaction. Suppose the microbes through the skin they enter, they give rise to the dendritic cells producing cytokines acting on neutrophils. They also activate the macrophages. They can also produce chemokines acting on monocytes, and they can also via lymph ducts travel into lymph nodes and interact with the T cells. Similar way, the dendritic cells also can travel under the stimulation of neutrophils and travel into lymph nodes to interact with T cells. So uh, so T lymphocytes have also got T cell antigen receptors or TCRs on their surface, similar to immunoglobulin, and TCRs can only recognize antigen with associated with MHC glycoprotein. CD4 T helper cells usually they usually respond to antigen presented by MHC class 2, and cytotoxic T cells or CD8 cell respond to antigens presented by MHC class 1. Once T cell binds to antigen, it triggers cell division to form both memory T cells and effector T cells. So that is how the T cells act. Apart from the T cells are also different subtypes, but two interesting subtypes to be mentioned is alpha beta T cells, which include CD4 cells predominantly and CD8 cells also, where there is a small subset between less than 10% gamma delta T cells which can present CD4 negative, CD8 negative in 60%, and only about 30% have CD8 positive marking cell surface markers on their surface. So it's interesting that intrauterine the gamma delta thymocytes are more predominant, but from about 16 days of gestation, slowly over about by 6 months, the alpha beta thymocytes predominate, and the gamma delta T cells drop down, and at birth they are very insignificant in number, as I have shown you. This picture is important to know how the T cell T helper subsets develop. So from naive CD4 T cells under the influence of IL-12, IL-18, and through this pathway of these genes, they produce gamma interferon, TNF, and IL-2 to promote autoimmunity, or Th1 pathway as it is called, or under the influence of IL-4, IL-6, and through the influence of STAT6 and GATA3, they become Th2 cells to produce IL-3, 4, 5, 9, and 13 to promote allergy, as in parasitic infestation and allergy. And under the influence of IL-1, 6, IL-23, and TGF beta, and through the actions of ROR gamma T as well as ROR alpha, STAT3, IRF4, and AHR, they become Th17 cells to produce IL-17A, IL-17F, IL-21, and IL-22 to promote autoimmunity and inflammation, and under the influence of IL-6 and BCL6 gene, they become T follicular helper cells to produce IL-21, IFN gamma, and TNF, and they help for follicular zone B cells. And TGF beta and FOXP3 gene and RUNX3 gene, they become T regulatory cells producing TGF beta, interleukin 10, and CTLA4, which suppress immune response, or they become so. Uh, T cell differentiation has two different types. Naive T helper cells usually get activated only by T cells, whereas memory T helper cells get activated by macrophages, dendritic cells, and B cells. So what happens is normally the antigen presenting cells and the T cell receptors, they interact in presence of the co-stimulatory signals which help in dividing or proliferating the cells several times a day, and they gradually populate to memory and effector cells after the antigen exposure. Uh, B lymphocytes are formed and matured in the bone marrow. They exhibit receptors on the membrane. Once the naive antigen, they divide rapidly to produce plasma cells or effector B cells or the memory cells. The NK cells are activated by viral or tumor antigens in affected cells. They recognize altered activating receptors and inhibitory receptors. They release preformed effector proteins from secretory granules, namely granzymes and perforins, and they kill altered cells by inducing apoptosis or by antigen dependent cellular cytotoxicity. So if you see the various cytotoxic cells response, the CD8 positive T cells and NK cell almost act in a similar fashion by producing perforins and interferon gamma, TNF, and fast ligand pathway, and they give rise to cytotoxic granule release and fast ligand interaction, leading on to onto cell death or mechanism of killing. Whereas NK T cells are a subset of cells which has got features of both NK cells and T cells. They produce IFN gamma, IL4, GM-CSF, IL2, and TNF, and they predominantly act by fast ligand interaction and can activate NK cell. This is an example in a graft versus host disease where the host cells, which is a transformed cell, in response to the graft, when there is a fast ligand pathway is activated and induce apoptosis in the graft versus host disease. Whereas in a virally or tumor infected cells, in cytotoxic T cells, when they encounter them, they produce perforin and granzyme, and they attack these cells and they induce host disease, graft versus tumor disease cell. So graft versus tumor mechanism tumor apoptosis is induced by different mechanism by the cytotoxic lymphocytes as compared to disease. So this is another important picture. I would like your attention here to have a look. If you look at the picture on the left side, this is a T helper cell and the antigen presenting cell. On the right side, T cytotoxic cells and the target cells. So if you see these two pictures, you can see that the LFA1, ICAM1, these are all addition molecule and the ligands interaction. We have the MHC class 2 presenting the external peptide and interacting with the CD3 TCR T cell receptors to the T helper pathway. As I have told you that the CD4 cells are important for MHC class 2 mediated response, and there is also CD22 CD45R interaction, and this last one is known as the co-stimulatory B7 CD28 interaction. So if you see this and this, if you compare, what you see is, except for the here it is CD4, it is CD8, and instead of class 2 it is class 1 MHC. But the major difference what you can see here is that here there is no co-stimulatory signal. So when cytotoxic T cells act on the target cells, whether tumoral cell or virally mediated cell, they do not need a co-stimulatory signal. Now this is interaction between the dendritic cell and T cells in a even more simplified fashion. So this is a class 2 MHC on a dendritic cell presenting the antigen, and they are being presented to the T cell and you can see that it can and there is a co-stimulatory molecule acting on, and depending on the cytokine milieu, see how they respond. They either go into a Th2 pathway producing IL4, or if there is a lot of IL12 and IL18, this can cause transformation to Th1 pathway producing interferon gamma, or it can become a Th17 pathway by producing IL17 under the influence of TGF beta, IL6 and IL23. So this shows how the same presentation can give rise to different type of transformation of T cells depending on the various cytokine milieu. So another important factor I would like to emphasize here is the co-stimulatory signal is very important, because in absence of co-stimulatory signal, whether if the B7 is absent or the CD28 is blocked by an antibody, or the T cell is turned into anergic some way, in absence of co-stimulatory signal, there will be clonal energy, or they will become ineffective. They will not produce to produce IL2 and T cell proliferation as would happen in a normal scenario. Now I have talked about immune organ and cells. Now we talk about immune molecules. Predominantly four categories of molecules we have to know: antibodies, complements, cytokines and interferons. The antibodies are immunoglobulins. They are similar to TCR on T cells. They are Y-shaped. They have got two heavy chains and two identical light chains, and these are the various 5 isotypes all of you know, from IgG to IgE, and they are basically, they belong to gamma globulin fraction in serum protein electrophoresis. What are the functions of antibody? This picture beautifully describes the major 6 functions of antibodies. Number 1, it can activate the B lymphocytes, producing more antibodies, and also stimulating them to produce memory cells and plasma cells and secrete further antibodies to kill bacteria. It can, excuse me, it can also act as an opsonins, so like a saponification, and it can enhance the binding bacteria to them, and cause enhanced phagocytosis, and that way it kills the bacteria. Or it can cause antigen clumping and inactivation of the bacterial toxins, and it can also give rise to antibody dependent cellular cytotoxicity. As I mentioned, sometimes NK cells act like that, and it can also trigger mast cell degranulation, releasing the granule components of the mast cells, and which are toxic to cells, and they can kill bacteria. Finally, it can cause activation of complement and thereby killing bacteria. So these are the 6 different pathways which are important for antibody function. The complement system, if I come to, there are 3 we know. Initiation of complement activation can be via alternate pathway, classical pathway and lectin pathway. In all the 3 pathways, they act on C3, and C3 becomes cleaved into C3b and C3a. And C3a is a pro-inflammatory molecule, so it induces inflammation. C3b is deposited on the microbes and opsonizes and causes phagocytosis, and C3b also act on C5 into C5b and C5a. And like C3a, C5a is also pro-inflammatory and induce inflammation. Finally, C5a also is important in the process of formation of membrane attack complex C5 to C9, which causes lysis of microbes as the late or the end stages. Simplified picture of complement. If this can be further simplified in this as you can see here, that in the classical pathway, where antibodies bind the microbes and the C1q C3 dividing into C3A and C3B, or in alternate pathway the microbes directly can act, and lectin pathway with MBL they can also trigger C3A dividing breaking up into C3A and C3B. C3B finally also helps in opsonization, again helps in phagocytosis and killing of bacteria, or it can also help in C5 splitting into C5A and C5B. As I told earlier, C3A and C5A both are pro-inflammatory, whereas C5B enhance formation of membrane attack complex causing lysis of bacteria. So this is how the complement system act. Now about cytokines, very important, know few basic facts of cytokines. Low molecular weight proteins they are, they bind receptors and they alter gene expression. They can be autocrine, paracrine and endocrine. It means autocrine means they can act on their own cells where they are produced from. Paracrine means they act on the surrounding of the cells. Endocrine means they act distant parts of the body by going through bloodstream. Cytokines also regulate the immune response, and they are pleiotropic in action. It means that they can be acting differently, having different effect on different cells at different sites. So, inducing cause cytokine gene activation in the cytokine producing cells, and that produces cytokines which act on the receptors, which causes intracellular signal transduction, causes further gene activation and leading on to biological products by producing the target cytokines. So this is how the cytokines act. Now comes interferons. Interferons are a group of signaling proteins. There are broadly three types of interferon. Very important in the COVID scenario, interferon. It can be type 1, interferon alpha and beta. Type 2, interferon gamma. And type 3 is interferon lambda. The interferons have the ability to interfere with viral replication, therefore the name. It also inhibits tumor growth. It activates immune cells such as natural killer cells and macrophages, and it also enhances T lymphocyte activity. So if you see this picture here, you can see that type 1 interferon and type 3 interferon through this various JAK and STAT pathways and interferon regulatory factors, it causes intracellular signal transduction, and it causes the antiviral activity. Whereas the type 2 interferon is important, they in terms of antimycobacterial activity. So you know type 2 interferon is gamma interferon, that's why you know that gamma is an assay for mycobacterial infection. So in the case of COVID, these are and type 3 interferon. Now comes to uh, the various ways by which primary induction of type 1 interferons happen. Type 1 interferon may be primarily induced by cell intrinsic inducers or secondarily induced by cell extrinsic inducers. Cell intrinsic inducers include RNA triggers, retrovirus, line 1 mRNA or ALU rich 3' UTR mRNA, RNA fragments, DNA triggers like reverse transcribed DNA, incomplete reverse transcribed DNA and dsDNA fragments. Whereas so these are products of internal cell death and cell activities in life, whereas extrinsic ones can be because of external factors like free chromatins, neutrophil derived mitochondria, neutrophil derived net or neutrophil extracellular traps formation, apoptosis or necrotic cell debris, and this all can happen in response to microbial process, nucleic acid containing immune complexes. All these things can trigger a secondary induction of type 1 interferon through TLR7 and 9 producing interferon alpha, whereas primary induction, as I mentioned, through the cell intrinsic inducers cause upregulation of these genes REG1, MDS and CGAS, and they produce interferon beta. Now comes to the effector immune functions. I talked about organs, cells, and the molecules. The functions. Effector mechanisms, immune responses. Before the immune mechanism, there are some natural barriers like epithelial cells of intestine, respiratory tract, the physical barriers like tight junctions or even the skin. The mucins, heavily glycosylated, prevent attachment of microorganisms. Defensins are molecules which are peptidic components cause direct killing. TLR5 on the basolateral surface of the mucosal cells, they actually so interesting that they cause TLR5 signaling to kill invading organism is so unique that they only respond if there are invading organisms there. They do not respond to luminal commensals. You know that human body or human tissue has got more microbial genetic material than us, than our own, and many of them over the evolution have become part of our DNA. So we have commensals or microbiota or gut flora, respiratory flora, genital flora. They are protective to us. The immune cells, especially the TLR4, do not aggressively respond to the normal bacteria, normal microbes. Then comes to intraepithelial T lymphocytes, which I mentioned to you in the when I've shown you the malt picture. They recognize glycolipids of microbes. Now, very briefly, just oversimplified version of innate and adaptive immune response. It's very interesting. Just if you have to remember by one or two words, what is innate immune response? It is like the reflex immune response. It is relatively few. It responds almost instantaneously. It has got evolution has taught the innate immune system how to respond aggressively to certain components of microbes like lipopolysaccharides, lipoteichoic acid, glycans, etc. And these will almost act immediately, and they are very perfect and very effective. And this is how they act within minutes to very fast they act. So this is innate immune response. Whereas adaptive immune response is like adult human beings. Give me a chance, I will learn more. Give me another chance, I will learn more. So adaptive immune system does the same thing. They are they are little slow, they are delayed, they are clonally driven, unlike non-clonal in innate immune system. They observe the the antigen response, and they carefully which pathway to go. So this comprises of the humoral and the T cell or the cell mediated arms of the immune system, and this is less perfect than innate immune response. It takes time, and it also but good thing about the adaptive immune response is it leaves behind a memory, so that next time that organism comes, it can respond back through memory cells and the effector cells. So whereas innate immune response do not have any memory. So the pattern recognition receptors for PAMP and DAMP, or pathogen associated molecular pattern, and death or danger associated molecular pattern. So these are, as I have told you, in innate immune response, how does the immune system respond so immediately, whether it is a lipopolysaccharide or not. So as evolution has taught our immune system certain things, that if sees it, it will attack. So there are two types of such proteins, pathogen associated molecular patterns and death or danger associated molecular patterns, and in response to them we have pattern recognition receptors. There are broadly three different types, or even little more types also are there: secreted PRRs, endocytic PRRs, and signaling PRRs. The secreted PRR act via complement activation and opsonization, whereas endocytic PRRs, they cause by pathogen uptake by phagocytes, and signaling PRRs cause activation of the inducible innate immunity. So this is about broadly the picture of how the PRRs act. The pathogen can give rise to a signaling PRR response through TLR and acting on the gene expression of the specific cytokines, or signaling PRRs can also act via cytosolic PRRs into the specific gene expression for specific cytokines to kill microorganisms. Internalizing PRRs actually act again through TLRs, but they get internalized by the pathogen and they produce the peptides and antigens which are presented through MHC, and again through that they are presented to T cells, which I have mentioned to you earlier. Or there may be soluble PRRs which can cause either protease cascade response to inactivate organisms, or they can opsonize and help in phagocytosis and thereby again internalizing it. So pathogen recognition methods by PRR again can be broadly classified by three mechanisms. One, non-opsonic recognition. I told you opsonization or phagocyte opsonization, like a saponification, and they bind the bacteria and they help in phagocytosis. Or that may be, this is this is opsonic receptors, through opsonic receptors, or it may be non-opsonic receptors where the serum opsonins bind to the membrane PRRs, and they get phagocytosed, and they inside the cell then they get degraded, and they are recognized by phagosomes, and microbes are killed, and the cytoplasmic recognition happen by NLR proteins or inflammasomes, or they can also act via TLR, producing inflammatory response. On the other hand, there can be also multiple recognitions. There can be both opsonic and non-opsonic recognition through the various membrane PRRs. So the PRRs can respond by opsonic, non-opsonic, or other mechanisms which eventually lead on to phagocytosis, degradation, recognition by inflammasomes, TLRs, and producing or presenting them through the MHC. So interaction of the antigen presenting cells with T cells, as I have told you, PAMs like lipopolysaccharides from pathogens, or endocytic PPRs like MRs, they get into the cell, and the antigen processing happen, and through MHC they are presented to T cells through to the T cell receptors, and then they give rise to production of various cytokines, or the signaling by PPRs and like TLR4, as I mentioned, they is internalized, and then they give rise to production of cytokines like pro-inflammatory cytokines like IL-1, 6 and 12. Whatever it is, the ultimate end result is to target the microbes, etc. Now, induction of inflammation therefore can be by TLR signaling, by internalizing PRRs, inducible nitric oxide synthase pathway, by NLR inflammasomes, or when very severe uncontrolled cause endotoxic shock. So in various pathways inflammation can be induced, and the activation of the adaptive immunity happens by antigen presenting cells at the site of inflammation. They mature and upregulate the chemokine receptors, they go to lymph nodes, activate T cells, they internalize PRRs, antigen presentation, they signal PRRs and enhance co-stimulatory molecules, or they TLR ligands activate antigen presentation producing Th1 or Th17 inducing cytokines. So these are the various ways the innate and adaptive immune system respond. This is a list of the TLRs or toll-like receptors from TLR1 to TLR10, out of which very important are 7, 8, 9 and 3, because they are endosomal, they are localized in the endosomal compartments, so they can respond to internal antigens, or they can respond not only to external one, they can also respond to internal antigens. So the type of lymphocytes are B cells, helper T cells. I have two more slides. B cells, T helper cells, cytotoxic T cells, regulatory T cells, and natural killer cells. What the B cells does? They help in antigen recognition. The antigen on pathogens, all soluble antigens. They produce antibodies. They neutralize pathogens. They phagocytose and complement activation. Whereas helper T cells, what do they do? They present antigens presented to them by professional antigen presenting cells are recognized. They cause secretion of cytokines. They activate macrophages. They activate other T and B cells and cause inflammation. Cytotoxic T cells, what do they do? Uh, they respond to again antigen presented by infected or malignant cells. Elimination of the infected and the malignant cells happen through them by interaction of the cytotoxic T cells. Regulatory T cells, no antigen recognition is present. So remember, these are immunosuppressive cells. When they regulate and suppress immune response. When regulatory T cells are overactive, there is a chance of bacterial infections because they suppress the normal immune response. At the same time, they actually balance or prevent autoimmunity. Natural killer cells, they want to self antigens or foreign antigens on host cells like virally infected cells, and they eliminate the infected and malignant cells. Here they eliminate the cells by direct action. So towards the summary slide or take home message is that thymus, bone marrow are the primary organs. Spleen, lymph node, organ. Neutrophils, basophils, eosinophils, mast cells, monocytes, dendritic cells, T cells, B cells, NK cells, immune system, and include immunoglobulins, cytokines, interferons, and complement. And the final effector function is the pathogen clearance, either causing autoimmunity or preventing autoimmunity, causing auto-inflammatory pathway. As I mentioned something about NLR or the inflammasome mediated, are called auto-inflammatory diseases like familial Mediterranean fever or even gout. The allergic response, the immunodeficiency, as you know, the primary immunodeficiency conditions, they act like nature's animal models. So the specific immune arm defect can give rise to a different specific type of immunodeficiency. And then there are cancers. You can have altered immune system causing immunomalignancies. Example, multiple myeloma, which is a plasma cell malignancy. So with all these things, I would like to conclude here. I tried to give an overview. These are the three my recommended YouTube videos you can watch. The first one is something like what I have talked. Second one is something meant for layman. And the third one is interesting. You have a picture of two photon microscopy. Two photon microscopy is very interesting. They have in vivo pictures of how dendritic cells, T cells, and B cell act with each other, or NK cells act with the antigen presenting cells or the target cells. It's beautifully seen in videos in real life. So this one is about 2 minutes. This is a little longer. So I request you to go through this. Thank you very much. I'm sorry for overshooting the time a little bit, but this was so. So without wasting much time, I would now like to move on to Dr. Professor Professor Abel Lawrence will talk to us without slide, and as I feel sometimes this no slide gives us more what should I say, more lucid understanding. So he will speak on how COVID evades and tricks the immune system. So this is something Abel Lawrence is mastered by reading whole lot of information, and he has been a public speaker to through social media, including CNN. So I request Dr. Abel Lawrence to speak on how COVID evades and tricks the immune system.
Abel sir, if you can please unmute your mic.
Okay. Somebody called SARS-CoV-2 an airborne HIV. At first, I was taken aback, but as I read more about this virus and how it hacks the immune system, actually, I'm quite impressed. In fact, HIV is a novice before this particular virus. In fact, if you look at the lifestyle of these coronaviruses, they jump species to species and they need all these immunosuppressive tricks. And there are several things odd about this virus when you compare with the usual other common viruses that we encounter. One, even in the presence of antibodies, this virus is able to infect people. That is, even when there's an antibody that blocks the spike protein, it can still evade and infect it if the sufficient viral doses is there. I will explain. I'll come back to this later. Then this virus has a relatively long incubation period. Although it can be as short as 2 days, it can be up to 2 weeks, and there are instances where up to 4 weeks has happened. How does this happen? That means this virus is multiplying under the radar without our immune system noticing it. And then even after the immune response is raging, there's a wildfire of immune response is raging, people continue to excrete the virus. And now there are reports of people having excreted the virus for many weeks and even months. There's a case of up to 6 months excretion. These are rare but not common, but it's possible. How is this possible? This virus persists despite all these things. And then uncharacteristically, this virus is able to induce a powerful, ineffective immune response with the production of so many cytokines, IL-1, IL-6, and which is doing a lot of harm. And finally, if there is time, I will try to link how aging and aging-related diseases like diabetes, hypertension, and inflammation wreaks havoc, how the virus co-opts these systems and causes further damage to the body. So to begin with, let us see what happens when a virus comes and infects. The first stage, this particular virus, the spike protein, which binds to the host cell receptors, actually is double-faced. Unlike a virus which has a cognate receptor, this can actually have two different receptors. There are two domains for the spike protein which can bind to two different receptors. So one of them in humans is ACE2, the other one is neuropilin-1. So this existence of two different receptors allows the virus to evolve independently on both of these. So it can bind to one receptor while the other one is varying, and then when it crosses species, it can use the other receptor while this one adapts. So this ability of two different receptors gives it an ability to evade host immune system, host, jump host, and actually gives it flexibility and adaptability. Then once the virus enters the system, our innate immune system is supposed to recognize the presence of the virus. As Dr. Danda pointed out, there are a whole lot of innate immune receptors, pattern recognition receptors, which detect pathogens. And there are a lot of viral sensing molecules. And a lot of there are toll-like intracellular toll-like receptors like TLR7, which can detect viruses. And then there are various large families of interferon response genes which belong to what is called RIG-like helicases, including MDA5, even TRIM21 can also recognize enterovirus, some of the viruses. So there are many proteins which detect presence of virus and activate, and these intracellular pattern recognition receptors, they activate interferon response. But characteristically, when you infect cells with SARS-CoV-2, they don't mount an interferon response. This is very strange. That is, infected cell lines infected with SARS-CoV-2 do not mount an interferon response until very late, until up to 50% of the mRNA inside the cell is of viral origin. So this virus can basically stop the dam as far as the innate immune sensing is required. And then another thing that happens is this induces production of IL-1 and then IL-6. How does that happen? So you know that IL-1 response comes from the activation of inflammasome. There are a whole family of cytosolic sensors called inflammasomes, and this virus activates it. I'll come to how this happens. First of all, let us say, how does this suppress interferon response? People have been looking at it, and not less than 8 viral proteins have been identified which can stop the interferon response. So in inhibiting interferon response, this virus has 8 different non-structural proteins which can do that, including ORF9b, ORF9c. You will lose count of that. Then if that is not enough, there are proteins like NSP15 which can inhibit double-stranded RNA, that is viral RNA. Double-stranded RNA is always viral origin. It inhibits the pattern recognition receptors which recognize double-stranded RNA. Then there is an NSP8 which inhibits MDA5. So there it has, in fact, in the beginning, people identified about a dozen non-structural proteins. Now the number of proteins involved in immunomodulation has more than doubled. So this has got a whole lot of receptor proteins whose sole job is to modify the immune system. And we are not even finished cataloging them. Then what happens when a virus is infected? What the virus virus actually hijacks the protein synthesis machinery. So most viruses have an ability to inhibit host protein synthesis, that is host translation of host mRNA. And NSP1, one of the viral genes, it is common to most viruses, and duly SARS-CoV-2 also does that. The idea is that virus can then monopolize the protein synthesis machinery. And then virus produces its first, it produces the replicase, the machinery needed to synthesize its copies, that is, more copies of itself. Then it produces various other things. And then once the virus protein synthesis starts, all cytosolic protein synthesized, they get degraded in the proteasome and then presented in MHC molecules. That is, peptides derived from proteins are presented on MHC molecules on the cell surface. So when viral peptides are presented on the cell surface, host T cells can recognize it. This is how cell-mediated immune response works. T cells cannot recognize intact antigens. T cells can only recognize peptides which are presented in the context of host MHC. Now, this SARS-CoV-2 has a protein called ORF3b, which literally shuts down MHC class 1 expression. That means when the MHC class 1 expression is shut down, there is no presentation of the host protein, the viral proteins, and the CD8 cytotoxic T cells cannot recognize this. So this is probably one reason why CD8 response is ineffective in. But if you look at it, the first protein synthesized is not ORF3b. So there is a small window from the viral infection to the shutdown of the MHC class 1 expression before which the CD8. So if a vaccine can mount a CD8 response against the early antigens expressed by this, it may be possible to stop the viral replication. But once that is crossed, for example, spike proteins are expressed much later. So therefore, say vaccine against spike protein may not be able to stop an early infection, which is one of the reasons why the vaccines will not be 100% effective. They will only be partially effective. There are more mechanisms. I will come to that later. So now we see that MHC class 1 expression is downregulated, and there are many mechanisms by which this virus can evade, the T cell response mounted to it and inhibit interferon response. Then how does it induce this IL-1 response? So obviously IL-1 response requires inflammasome activation, and now they have identified a viral protein called ORF3a, which has been named viroporin. In fact, viroporin family of proteins are key mediators of viral infection-induced inflammation. And this viroporin can activate NLRP3 inflammasome and produce IL-1 response. So, and this, how does it do it? It interacts with a protein called TRAF3 and then interacts with NLRP3 inflammasome. So we know that we have a drug in our armamentarium which inhibits NLRP3 inflammasome. Anybody has an answer? Colchicine. In fact, colchicine inhibits NLRP inflammasome. There are studies which show that colchicine given to patients with COVID can reduce inflammatory mediated production, various cytokines. In fact, all the inflammatory cytokines and everything is inhibited by colchicine, and colchicine is a potential treatment option in, especially in people who have persistent inflammation and decrease in lung function. But it needs larger studies. The current studies available are small. I looked at secondary out measures like cytokines, and we need to look at hard endpoints. Then comes the how does and then I said that once immune system starts seeing the virus, an immune response is mounted, and you produce CD4 and CD8 lymphocytes and also antibodies. And then what happens to these antibodies? See, when generally it was earlier believed that when a virus is coated by an antibody, the virus is unable to enter the cell. This is actually not true. Actually, even when the presence of antibodies, lot of viruses manage to get through inside the cell. But still, it has been shown that even in the presence of antibody, even if virus enters the cell, in most of the situations, the virus cannot infect the cell, and the mechanism was not clear earlier. Now that has been understood. There is a mechanism called antibody-dependent intracellular virus neutralization. That is, when a virus enters a cell, it injects its material into cytoplasm and then takes over the protein synthetic machinery. And so the thing is, when an antibody-coated virus enters a cell, there is a protein viral sensor which belongs to the helicase family as well as the E3 ubiquitin family protein, which we are all familiar by another name. Most of you would know its name if I told its synonym. This protein is called TRIM21, which is also called Rho52. And Rho52 is one of the most common autoantigens in connective tissue disease, and it is present along with Rho60 in lupus, Sjogren's syndrome, and in myositis patients they find isolated Rho52. This Rho52, also called TRIM21, is the most important intracellular sensor of antibody-coated viruses. So what it does is, when it detects a antibody-coated virus, what it does is it ubiquitinates. And what happens when you ubiquitinate it? That ubiquitinated proteins and structures, organelles, they go into the proteasome. So basically, TRIM21 can short circuit the virus plants and rapidly induce proteasomal degradation of the viral proteins, and then they get presented on MHC class 1 in an accelerated manner. So this mechanism is very important for two classes of viruses, enteroviruses and adenoviruses. And in the case of enteroviruses, TRIM21 is not only a sensor of the antibody, it can directly sense some of the enteroviral proteins like the chymotrypsin-like protease. C3 protease of enterovirus can be directly sensed by TRIM21. And in fact, to prove this, mutations in the enterovirus 71 C3 protease, which evades binding of TRIM21, leads to more invasive central nervous system infection with enterovirus 71. So mutation of the target is a proof of that it is the target. And adenovirus also, this mechanism is very, very important, and which is important now regarding the the adenovirus is one of the vectors for coronavirus vaccine, and which is important now in the presence of antibodies, TRIM21 will effectively destroy the adenoviral vector. So which is why subjects who have antibody to the adenoviral vector will have a difficult time mounting an immune response, and this could be a reason for variability of immune response to adenoviral vector-based vaccines, especially in healthcare workers who are more likely to be exposed to a wider variety of viruses and may have antibodies to this. So now we can see that this particular virus can evade when an antibody-coated virus, but this evasion through and how, again, I didn't mention how does it happen. So the thing is, TRIM21 removes antibody-coated virus to ubiquitination, but the papain-like kinase a papain-like protease of the SARS-CoV-2. There are two main proteases in the coronavirus genome. That is the chymotrypsin-like protease and the papain-like protease. This papain-like protease has the ability to remove ubiquitins. So when TRIM21 adds an ubiquitin, papain-like kinase duly removes it. So once papain-like kinase removes the ubiquitin, virus escapes the So this is why coronaviruses as a class can escape antibody protection. But this kind of escape is not 100%, it's partial. So effectiveness of antibodies will also be partial. So this can evade cell-mediated immune system and also antibody-mediated immune system. And it can also activate IL-1. How is IL-6 produced? IL-6 comes downstream of IL-1. IL-1 can induce IL-6 production. But there are other mechanisms by which IL-6 can be produced. For example, when you have hypoxia, in say, let's take which is the best model for hypoxia? High altitude pulmonary edema or mountain sickness. One of the biomarkers of high altitude pulmonary edema and mountain sickness is actually CRP and IL-6. So when people are exposed to low oxygen situation, when the lung is hypoxic, lung produces both IL-6 receptor as well as IL-6. And this it takes about a few days for IL-6 production to increase in the lung in a hypoxia scenario. So again, you know that the inflammation comes a few days after the patient develops symptoms. So IL-6 production can occur from hypoxic lung. Then when somebody is hypoxic, they are under severe stress. And there are mechanisms for sympathetic nervous system induced active production of IL-6 from adipose tissue. And this happens in browned adipose tissue and is in response to stress, and normal selective beta blockers can't do anything because this is mediated with beta-3 adrenergic receptors, which selective beta blockers do not block. So increased sympathetic activity, like a fright response in COVID. And we know that when patients with COVID, what is the natural history? Patients develop symptoms. Initially, it is mild. They think everything is fine. Then suddenly, after 3 to 4 days, patients deteriorate. And some patients have several bouts of this deterioration. And during this deterioration, you might have noticed they are on hypertension. And so hypertension is also a sign of stress induced. So there is a considerable, when they develop stressed, there's hypertension and there's sympathetic activation, and there is renin-angiotensin system activation. So stress itself can produce IL-6. Then what is this connection between renin and angiotensin? There was initially a lot of fears about use of ACE inhibitors. That was due to a confusion. This virus uses ACE2, not ACE1. And ACE2 actually has a negative regulatory role actually in the renin-angiotensin system. So this ACE2 is usually found in association with the angiotensin 2 receptor, primary angiotensin 2, which is the AT1 receptor. Why is it found together? So when the angiotensin 2 binds to the angiotensin 1 receptor, which is the receptor for angiotensin 2, the adjacent ACE2 inactivates it by removing amino acids from it. And this inactivation leads to stoppage of further angiotensin signaling. So in the when there is similarly, when you have activation of the angiotensin receptor, that leads to internalization of both angiotensin 1 receptor and ACE2, leading to degradation, cleavage of the ACE2. Then the soluble ACE2 appears in the serum. So when there is an angiotensin activation, there is increased soluble ACE2, but the surface ACE2 comes down, and therefore the ability for further down regulation is reduced. And what happens is that once coronavirus infects some tissues, there is increased renin-angiotensin activation due to the loss of the inhibitory activity of ACE2. And then But the problem is that ACE2 is the viral receptor. If we have more ACE2, there can be more viral infection, but ACE2 is also needed for blocking renin-angiotensin activation. What are the things which upregulate ACE2? Inflammation and any kind of inflammation. In fact, if there is reflex, if you instill dilute hydrochloric acid into the bronchial epithelium, the expression of ACE2 in the bronchial epithelium increases. So somebody with, you can expect someone with gastric reflex who has microaspiration may be more susceptible to a coronavirus infection. We don't know what there's no studies, but we know that obese people are more susceptible and obese people are more reflex. But what else? Angiotensin activation in peripheral tissues also mediates insulin resistance. And increased angiotensin signaling can lead to increased insulin resistance. And it is not uncommon to see patients coming with coronavirus infection to have increased blood sugars. Of course, nowadays with a lot of steroids, that's an additional factor. But newly diagnosed diabetes is not, and we have seen patients with difficult to control diabetes along with this. Then people with hypertension, kidney disease, they all have naturally increased renin-angiotensin system activation, and this increased renin-angiotensin system activation will exacerbate the effect of the renin-angiotensin system activation. And there are inflammaging in blood vessels. There are studies where if you isolated blood vessels, if you give angiotensin 2 alone, nothing happens. But if you give senescent T cells, nothing happens. You combine senescent T cells and angiotensin 2, the vascular aging gets accelerated. So vascular aging processes get accelerated by increased angiotensin 2 and senescent T cells. And when your older people have more senescent T cells, and increased renin-angiotensin activation leads to more endothelial and vascular damage. Then another mysterious thing that associated with coronavirus is the increased thrombosis. So how can we explain thrombosis? One, this virus infects the endothelium, there is endothelial activation. And besides that, this, when I said deficiency of ACE2 leads to increased angiotensin 2 signaling, but there is something else also happening. Angiotensin 2 has two pathways of degradation. One is through ACE2 carboxypeptidase activity and through the aminopeptidase. And aminopeptidase activity on angiotensin 2 leads to the production of what is called angiotensin 3 followed by angiotensin 4. But if both carboxypeptidase and the aminopeptidase act, it leads to degradation and inactivation of angiotensin. So when you have a deficiency of angiotensin 2, but continued activity of aminopeptidase, for the deficiency of ACE2 and continued aminopeptidase, you have an accumulation of angiotensin 4, and angiotensin 4 has various effects. For example, increased expression of angiotensin 4 in the brain leads to hypertension, which is again one of the manifestations of sick COVID patients. That is, patients without history of hypertension present with hypertension in the emergency when they have COVID, at least the ones who are serious. And this actually has been shown to be blocked by angiotensin receptor blockers. That is because angiotensin 4 induced hypertension is due to increased angiotensin 1 receptor signaling. And then angiotensin 2 and angiotensin 4 both upregulate the production of plasminogen activator inhibitor 1. And plasminogen activator inhibitor 1 is a serpin that is, which increases, it's a procoagulant protein, and it inhibits most important anticoagulant proteins in the body, like such as tissue plasminogen activator. It inhibits protein C proteinase system. In fact, protein C proteinase system and this is antagonisms, they can inhibit each other. And this is something which is necessary and sufficient for thrombosis. Why I'm saying is, you might have all heard of seronegative antiphospholipid syndrome. In seronegative antiphospholipid syndrome, patients have clinical syndrome of antiphospholipid syndrome, arterial and venous thrombosis and pregnancy losses, but no anticardiolipin antibodies. In fact, there have been cases of seronegative antiphospholipid syndrome where they could not find any autoantibodies, but they found unusually increased plasminogen activator inhibitor 1. That means increased plasminogen activator inhibitor can itself cause thrombosis, and there is a natural diurnal variation in PAA1. It is more in the mornings and low in the evenings. And when does most thrombotic disease occur? Early morning, when the PAA activity is highest. So PAA, and in fact studies, this was some prediction we made in March, April, but subsequent studies have shown that PAA1 levels are increased in coronavirus infections. Increased PAA1 with the endothelial infection and endothelial activation can explain the increased thrombotic risk in patients with coronavirus. And even in diabetic subjects have naturally increased levels of plasminogen activator inhibitor 1. So basically, this virus has multiple mechanisms to evade immune response and evade both innate and adaptive immune response, and also hijack the immune system in such a way that there is significant amount of inflammation, and this interacts with damaged pathways in hypertension and diabetes through renin-angiotensin system, and worsens the outcome. And interestingly, among the things that decrease plasminogen activator inhibitor 1 include vitamin D. But unfortunately, this inhibition only is in the vascular smooth muscle, not in the vascular endothelial cells. So in vitamin D deficiency, you may have increased plasminogen activator inhibitor 1, but giving vitamin D in people who are sufficient, it may not do anything more. If anybody has any questions, it will be, I can answer.
Thank you, Dr. Abel. You have seen how beautifully he has explained the subject of COVID. He's one of the most confident in COVID science in India today. I have gathered it over the last 6 months. One day he spoke almost 1 hour to me on telephone, and I was impressed by the depth of knowledge he has, the amount of reading he has done. Really salute you, Abel. We'll take the questions at the end, but I cannot. There's one comment for you. Novel information: why obese and elderly are more prone for complications, why thrombosis is more in the morning. Excellent presentation. So you have got compliments, no questions as of now. The next presentation will be with slides. That is with Dr. Vishad Vishnath. He is probably taking the same thing what Abel has told in a more clinical perspective. Dr. Vishad is also another brilliant immunologist and rheumatologist. He is speaking now on how does COVID cause immune mediated injury, and I think that's very important. Please pay attention and listen to him, and keep typing your questions on the chat box. Welcome, Dr. Vishad. Dr. Vishad is the director of the IRIS at Trivandrum, and I am very fortunate to have worked with him as a colleague at Vellore for some time. Thank you. Welcome.
Thank you, sir. First of all, let me express my gratitude to Dr. George Chandy sir and Dr. Danda sir for giving me this opportunity. Secondly, I'm afraid that Dr. Abel has covered too much. Yes. Yes, intruded into my territory and probably has covered much of what I wanted to say. But anything, I think I'll anyway, I don't think there'll be any harm in repeating at least some of whatever has been already discussed for better comprehension. So as we know that the COVID-2 is associated with, generally it causes a very mild infection, about 80% of people. It just causes some mild respiratory symptoms, fever or cough or sore throat. And the virus enters, it has been found that there are different possible portals of entry. It can enter through the tongue, nasal mucosa, and possibly conjunctiva. And the virus can replicate in many of these areas, including the throat as well as the bronchial epithelium. Usually after infection, the peak viremia occurs in about 3 to 5 days. And most patients become asymptomatic in about 6 to 10 days. And it is only in about 20% of patients who progress into more serious manifestations. And I think we are all aware of what is the most serious manifestation, which is the COVID-induced lung damage. So it varies from mild inflammatory infiltrates to happy hypoxia to diffuse inflammatory, diffuse ground glassing, and even fibrosis in the very late stage. So I'll start with what is the pathology of in COVID in various tissue and predominantly in the lung. So what people have found out is that when they examined the alveolar epithelium in patients who had severe COVID, what they found was that there was extensive necrosis of the alveolar lining and there was diffuse alveolar damage. And because we learn once again, we learn later that the receptor for COVID is angiotensin converting enzyme 2, which Dr. Abel has already discussed, and that is the type 2 pneumocytes in the lungs which have the maximum expression of this particular enzyme receptor, and hence binding of this enzyme receptor to the COVID in the type 2 pneumocyte can lead to pneumocyte hyperplasia, but at the same time this becomes fairly dysfunctional as well. So what it ultimately leads to is that there is extensive homogeneous fibrin deposit, interstitial edema, and what is important is that there is early organization of the inflammatory infiltrate. It is the inflammatory infiltrate gets organized faster as compared to many other pneumonia, and this leads to significant impairment of the gas exchange. So basically what happens is that there is destruction of the alveolar lining, there is extensive inflammatory infiltrate as well as fluid-like fibrin-filled deposit which occurs into the alveoli, which leads to which organizes itself and leads to early impairment of the gas exchange. And one another finding in the lung biopsies in patients with COVID has been the extent of endothelial damage which has been noticed. So the endothelial injury and disruption of endothelium has been fairly well documented. In addition to the endothelial injury, what has also been peculiarly noticed in COVID, as Dr. Abel mentioned, is the presence of vascular thrombosis and microangiopathy and occlusion of the alveolar capillaries. In addition, because of the severe extent of the vessel damage, there is also an attempted reparative process leading to new vessel formation. So basically, the pathology in COVID happens begins in the epithelium and extends to the endothelium, and it leads to epithelial damage. It leads to endothelial disruption, and ultimately leading to thrombosis. And it is not just the lungs that are involved in COVID, especially in severe COVID. The other organs are also very well documented to be involved. For example, anosmia and algesia are a couple of findings which are seen in central nervous system. So in CNS, it has been found that it can lead to edema and degeneration of neurons. It can involve the heart. In fact, cardiac manifestations are one of the most important cause of mortality in COVID. So it can lead to edema of the cardiac myocyte, atrophy of myocardial fibers, and it can lead to inflammatory infiltrates. These are all documented in patients with COVID in the autopsy studies. And of course, as we discussed in the blood vessels, it can lead to coagulation, activation, microcirculation disturbance. Another major organ which is involved in COVID is the intestine, and diarrhea is one of the common manifestations of COVID. And in intestine, focal inflammation has been documented, and even as Dr. Abel mentioned, diabetes is one of the manifestations of COVID. Although it could be related to inflammatory insulin resistance, there is also a possibility that it could be because of the islet cell damage incurred following COVID infection. Now, what determines this pathology in COVID? So when we are looking into the pathogenesis of COVID infection, there are two main mechanisms which are in operation. One is the activation of the angiotensin converting enzyme 2 and activation of the renin-angiotensin axis. And second are the immune abnormalities which are peculiarly seen in COVID. So as we already discussed, the portal of entry for the virus into the human lung epithelium or the respiratory epithelium is the receptor enzyme known as the angiotensin converting enzyme 2, which the enzyme per se is actually located in the X chromosome. It is the protein is coded by the X chromosome. And incidentally, this is also the receptor for the SARS-CoV infection, which we are all familiar with. And contrary to what happens in SARS-CoV-1 or SARS-CoV, this SARS-CoV-2 has a much higher affinity for ACE2 enzyme, which could explain the high level of infectivity which is associated with COVID-2. So the higher the affinity, the chance of getting infections are higher. So following infection, the COVID binds by means of the spike protein to the ACE2 receptor, and then it is cleaved by this one of the protein cleavases, such as the TMP-RSS2, which leads to further binding of this COVID virion to the surface of the epithelium, and finally internalization of this virus. So what determines which organs or what are the clinical manifestations? So it is basically, if we look at this, the clinical manifestation can very well be explained by the extent of ACE2 expression in various tissues. So ACE2 is highly expressed in tissues such as lung and intestine. In addition, they are in vasculature as well. In addition, they are expressed in heart, eyes, central nervous system, kidneys, gut, etc. So it is fairly ubiquitously expressed, and the expression of this enzyme receptor can explain much of the pathogenesis which are associated with the tissue trophism of the in the pathogenesis of the virus. So just to make clear what this how this angiotensin axis get activated in or the interaction of this angiotensin activation axis following infection by the SARS-CoV-2 onto the AC receptor. So we know that there is a peptide called angiotensinogen 1, which is converted to angiotensin 2 and by the renin, and which is then converted to by the angiotensin converting enzyme into angiotensin 2. So this is the normal, the usual metabolism of angiotensinogen to the usual pathway for formation of angiotensin 2. As we know, angiotensin 2 is a pro-inflammatory mediator. Now we also have this other enzyme which is known as the angiotensin converting enzyme 2, which what this angiotensin converting enzyme 2 does is that it converts the angiotensin 2 into another chemical which is known as angiotensin 1 to 9. In addition, it can also convert the angiotensin 1 to another chemical which is known as angiotensin 1 to 7. So what is common to this angiotensin 1 to 7 and angiotensin 1 to 9 are that these have quite anti-inflammatory property as compared to the pro-inflammatory property which is associated with angiotensin 2. So the formation of angiotensin 1, while the angiotensin 1 is pro-inflammatory, it can cause oxidative stress. It is pro-fibrotic, pro-thrombotic, anti-fibrinolytic, cell proliferation, and can lead to vasoconstriction and lead to cell injury. So an excess of angiotensin 2 can be expected to cause any of tissue injury by any of these mechanisms. Whereas the angiotensin 1 to 9 and angiotensin 1 to 7, the angiotensin 1 to 7 particularly, because this has more trophism for the angiotensin converting enzyme 2. So angiotensin 1 to 7 acts via the MAS receptor, and these have cell protective action by means of anti-inflammatory, antioxidant, anti-fibrotic, anti-proliferatic, and anti-thrombotic actions. So what happens following an infection? In addition to the angiotensin pathway, the other pathway where the angiotensin converting enzymes have an important role to play is in the tissue bradykinin pathway. As we know, bradykinin is a pro-inflammatory cytokine, and it mediates inflammation, can cause vasodilatation. So what happens is that the angiotensin converting enzyme 2, it is also involved in the degradation of bradykinin to inactive forms, thus leading to, and this leads to another anti-inflammatory effect in relation to the angiotensin 2. So we know that bradykinin is a pro-inflammatory cytokine and pro-inflammatory mediator. So what happens is that angiotensin converting enzyme 2 is involved in the, usually it is involved in the degradation of bradykinin to inactive forms. And in the absence of angiotensin converting enzyme 2, what we can expect is that there is unopposed action of bradykinins or other pro-inflammatory derivatives of bradykinin on the bradykinin receptors. And this can promote inflammation in the local area where it is, where the expression is occurring. So what happens when when the COVID infects the angiotensin converting enzyme, it binds with the angiotensin converting enzyme, is that along with the COVID, the angiotensin converting enzyme 2 is either internalized or it is shed into the shed from the epithelium. So what happens is that there happens a deficiency of angiotensin converting enzyme 2 on the epithelium. So what this ultimately leads to is an unopposed action of angiotensin converting enzyme and increased production of the pro-inflammatory angiotensin 2, which can lead to the other pro-inflammatory and pro-fibrotic actions. So what happens, again, I would like to reiterate this point. What happens is that on the binding of the virus with the receptor, the receptor virus complex get internalized, or the receptor can get shed from the surface of the epithelium or the endothelium. And so there is a local inavailability of angiotensin converting enzyme 2, which usually holds the angiotensin, keeps the angiotensin 1 levels and activity under check. So what it ultimately leads to is an unopposed action of the angiotensin 1 and 2, leading to the pro-inflammatory outcomes. And even before the onset of the COVID-2, experimental people had looked into the role of angiotensin converting enzyme 2 in lung injury, and they have very nicely demonstrated that in animal models of acute lung injury, if you knock out the angiotensin converting enzyme 2, the lung injury gets exacerbated. And to reiterate the fact that the angiotensin converting enzyme 2 knockout lung injury is mediated by angiotensin converting enzyme 1, people tried to knock out both ACE2 and ACE1, and what they found was that when you knock out both, the lung injury is ameliorated. Again, reiterating fact that it is not that angiotensin converting enzyme itself 2 itself is protective, but the angiotensin converting enzyme 1 leading to excess formation of angiotensin is harmful. And this also opens a therapeutic avenue, because one of the promising methods that have been tried is the use of recombinant angiotensin converting enzyme 2, which can potentially counteract the acute lung injury. So as we have just dealt with the first half of the pathogenesis, that is how the angiotensin axis gets activated when COVID binds with the angiotensin converting enzyme 2. The second half, I'll deal with the interaction between the immune system and COVID. If we sum up the interaction between immune system and COVID in 2 ways, in 2 sentences, this is what happens. There is hyperstimulation of the innate immune system, and at the same time, there is suppression of the adaptive immune system. So how this happens, we'll just go through in the next few slides. As Dr. Abel mentioned, one of the very interesting clinical features which are associated with COVID infection is the lesser expression of interferon 1 and interferon 3 as compared to other respiratory viruses. As we know, interferon is a very important mediator it's the first line of defense against viruses, viral infections. So probably because of the fact that this virus has interferon suppressive mechanisms, various interferon suppressive mechanisms by virtue of production of non-structural proteins or even structural proteins. By some mechanism anyway, this virus is able to suppress the expression of interferon in the affected tissue. So what this ultimately leads to is that this delays the viral clearance. So this gives the virus adequate chance to multiply to large numbers before that is cleared from the epithelium. Another finding, usually when there is a viral infection, there is on one arm you have the interferon activation, on another arm you have the cytokine activation, that is the NF-kappa beta mediated cytokine activation. So what happens in COVID infection is that although the first half, the interferon activation is not that optimum, The cytokine profile, a cytokine profile which is similar to hemophagous lymphocytic syndrome, happens and leads to increased production of IL-2, IL-6, GCSF, IP-10, MCP, and MAP-1 alpha. So these cytokines are extremely pro-inflammatory. And how is that these cytokines are produced? Different mechanisms have been suggested. One is that it is possible that there is a direct interaction of the non-structural protein 9 and 10 with the NF-kappa beta repressor protein. And this leads to an XU because the repression of NF-kappa beta is relieved. And as we know, NF-kappa beta is the master regulator of inflammation master regulatory transcription factor of inflammation in the cell. So because this NF-kappa beta repressor protein is inactivated, there is exuberant activation of the innate immune system, leading to extremely high levels of production of these pro-inflammatory cytokines. Another finding that has been seen in patients with COVID in the pulmonary epithelium, as Dandasar had mentioned, is the neutrophils and neutrophil activation and what's this process which is known as netosis. This is another important finding, which is netosis is another important finding which is seen in the epithelium of patients with subjects with COVID-2. In addition, there is also macrophages are also thought to have an important role. Macrophages have receptors for the angiotensin converting enzyme 2, and hence it is possible that COVID-2 can directly infect the macrophage, and this direct infection can lead to activation of pro-inflammatory macrophages, leading to the hypercytokinemia. And there is adequate evidence in the form of determination of detection of expression of spike protein on macrophage surface and expansion of pro-inflammatory monocytes and monocyte-derived macrophages in the lung, indicating that there is definitely a very important role for macrophages and the interaction between the COVID-2 and macrophages in the pathogenesis of COVID. And what is cytokine storm? What happens is that there is high levels of this pro-inflammatory cytokines such as TNF-alpha, interferon alpha beta, IL-6, IL-1 beta, etc., which ultimately leads to cell damage and cell apoptosis, leading to the multitude of severe manifestations which are associated with COVID. So this is how we can. This is how the innate immune system gets activated in COVID. While the innate immune system gets activated in COVID, the adaptive immune system goes into a state of slumber. So what happens is that what has been found is that in patients with COVID, there is the studies done on the epithelial washings and on the epithelial on the histology. What they have found is that there is decreased T cell activation and function of different forms of T cells, particularly the CD8 and NK cells. And it is what happens also is that the T cells just get exhausted, and there is increased apoptosis of T cells in patients with COVID-2. And this has to be interpreted in the fact that the T cells do not harbor much of ACE2 enzyme. Unlike macrophages, which harbor ACE2 enzyme, the T cells do not harbor much of ACE2 enzyme. So it is not likely to be a direct effect of the COVID-2 on the T cells, but it's likely to be mediated by the activation-induced cell death. That is, because of the cytokine storm, the T cells probably get overactivated and they get exhausted, and they stop functioning the way they are supposed to do. And another finding that has been very prominently noticed is that the adaptive immune response in COVID-2 seems to be polyspecific. Now, usually when we have an adaptive immune response, because it's a very robust immune response, the immune system has opportunity to mature itself to the antigen. The pathogen usually presents an immunodominant antigen, and the immune system tries to attack this immunodominant antigen with as much specificity and as much affinity as possible. So usually for infections, the adaptive immune response is tuned towards recognizing the immune dominant antigen. That is, it increases the chance of detecting the organism and killing it more effectively. If you have an immunodominant antigen to which the adaptive immune system is working, this immunodominant antigen triggers the immune system in a more effective way, ensuring that protein organism which happens that immunodominant antigen is cleared more effectively. But in COVID-2, what has been found is that the immune response is much more polyspecific. There is no single immunodominant antigen. While it is true that much of these antibodies are the T cell responses against spike protein, a fair amount of immune response is also mounted against nucleocapsid protein as well as the other non-structural proteins which are seen in COVID-2. So this indicates that by some mechanism, the COVID-2 escapes that process of somatic hypermutation and antigen and affinity maturation, which is important for generating that generating that high affinity and high killing ability towards that immunodominant antigen in a particular pathogen. Another important aspect regarding the adaptive immune system is another potentially important aspect is the role of antibodies in the disease pathogenesis. One observation has been that there is a higher titer of antibodies in patients with more severe disease as compared to patients who have a milder disease. So patients with COVID-2 who have a more severe disease, they have a higher titer of antibodies, including IgA, as compared to patients who have milder disease. And the antibodies also disappear much faster in patients with milder disease. In fact, majority of them clear off the antibodies in about 4 months, at least according to some of these studies. Now, this could either be the fact that the body's immune system is trying to resist against this, mount a very, very vigorous immune response against this in order to clear this severe infection, or alternatively, it could also mean that this higher antibody itself could be one of the cause for the severe manifestations seen in COVID. As Dr. Abel mentioned, it could be that these antibodies, when they bind, usually we expect that when the antibodies bind with the viral protein, they are internalized and and the viral proteins are selectively destroyed. But it seems that by means of it seems that one of the possible mechanisms that we can have here, similar to what happens in dengue antibody, is that the binding of this antibody with the virus could internalize it, and without destroying the virus, it could actually release it and make it permit it to proliferate in a much better way. So it is possible that antibodies may not need not necessarily be having a protective role. It could even be having a permissive role for viral replication in COVID. Another important component of the pro-inflammatory pathway associated pathogenesis associated COVID is the role of complement pathway. So we have the classical lectin pathway and alternative pathway. And COVID proteins have been associated, the various proteins which are present on COVID, they have been able to, it has been associated with activation of lectin pathway. They can potentially cause activation of lectin pathway. There is increased complement components, especially the terminal complement components have been noticed in the lung epithelium. Similarly, upregulation of complement proteins are also seen in sera of patients with COVID. A nucleocapsid, as I mentioned earlier, which is present on the COVID surface, can directly trigger the MPL or the lectin pathway. And it is possible that the IgE and IgA act in synergy to activate the antibody-dependent cellular cytotoxicity mediated by the classical complement pathway. So it seems that the pro-inflammatory effect of complements is another important arm in the pathogenesis of severe COVID. And we have seen that in the pathology that endothelial activation is one of the endothelial disruption, and procoagulation is one of the pathologies which are seen in COVID. How does this happen? So one is by mechanism of activation of the renin-angiotensin system, and we mentioned that activation of renin-angiotensin system can lead to unopposed action of the angiotensin 2, which is a pro-inflammatory, pro-fibrotic chemical, and it can also lead to activation of increased levels of bradykinin, which increases vascular permeability, and endothelial can lead to influx of pro-inflammatory cytokines and pro-inflammatory cells and lead to further damage into the endothelium. Activation of complement, macrophage activation, loss of endothelial integrity, and activation of neutrophils. These are the other factors which are associated with endothelial activation in COVID. And if we look at how this happens, so this is the what we have here is usually is a balance between the antithrombotic and prothrombotic signaling. In the absence of following internalization of the angiotensin converting enzyme 2 following binding with the SARS-CoV, what happens is that there is the balance tilts towards the prothrombotic signaling. This leads to increased production of bradykinin as well as angiotensin 1, angiotensin 2. And what this bradykinin and angiotensin 2 can lead is that it can lead to activation of the tissue kallikrein and the tissue plasminogen pathway or the procoagulant pathway. Another mechanism is by means of the complement. We mentioned that the complement activation is a feature which is seen in severe COVID-2, and complement activation can lead to again insertion of this membrane attack complex, which leads to endothelial damage, endothelial disruption, increased endothelial permeability, increased cellular infiltration, and the endothelium ultimately becomes leaky. In addition, the activated macrophages, they also destroy the endothelial glycocalyx, which is a protective coat on the surface of endothelium, which prevents thrombosis. And because of activated macrophages, this endothelial glycocalyx gets degraded. It leaves the endothelium raw and forms and permits it to form a nidus for thrombosis. The endothelium, the cytokines which are produced by the activated macrophages, the IL-1, IL-6, TNF, these are pro-inflammatory cytokine. And these again lead to endothelial activation and expression of pro-inflammatory and procoagulant proteins on the surface of the endothelium. It leads to increased production of adhesion molecule, increased influx of neutrophils. The neutrophils binding with the adhesion molecule further perpetuate the damage. So ultimately what happens is that the endothelium, following secondary to endothelial damage, the endothelium loses its anticoagulatory property, and it becomes procoagulatory, and ultimately leading to formation of activation of platelets and formation of fibrin thrombin. So now, to sum up, what happens in COVID infection is that most of infections, as we mentioned, are mild. And what happens is that the SARS-CoV-2 virus infecting the alveolar type 2 alveolar endothelial cells, in the presence of a mature immune response, there is the adaptive immune system is effectively recruited, and it comes and clears away the COVID virus. The virions are cleared away by means of CD8 CD8 cell activity or NK cell activity, and there is usually a resolution of infection and immune response, and finally the infected cells get apoptotic. So this is what happens in a usual, in 80% of patients, this is what would be happening. But in some patients, these disease can be very severe, such as in people who are older age, male sex, obesity, having diabetes mellitus, malignancy, and to lesser extent hypertension and COPD. And these are factors which are associated with various forms of immune dysregulation, and these are also associated. These could be also associated with the variations in ACE2 expression. For example, as Dr. Abel mentioned, as you age, the ACE2 expression on the cells reduces. Diabetes also been found to be associated with reduced ACE2 expression. Obesity, it has been found that the ACE2 is present in visceral adipocytes, and excessive activation of this ACE2, the angiotensin receptors are present in the visceral adipocytes, and excessive activation of angiotensin can lead to activation of visceral adipocytes, leading to a pro-inflammatory, leading to production of a pro-inflammatory milieu, thus perpetuating the inflammation in COVID. But what happens is that when in the presence of these risk factors, or in the these named risk factors, or the so many other risk factors that we are still not aware of, what happens is that the adaptive immune system is not able to respond adequately. The cell damage gets perpetuated. There is severe damage to the alveolar epithelial cells. The alveolar epithelium loses its integrity. The endothelium gets damaged. There is increased permeability and immune cell seepage, leading to further perpetuation of immune activation. The T cells at the same time, because they become apoptotic or they become exhausted, they do not respond as well as they are supposed to do in the face of this extensive inflammation. And this leads to unregulated action of this cytokine, pro-inflammatory cytokines and pro-inflammatory cells, ultimately leading to the disease pathology in different organs. Thank you.
Thank you, Dr. Vishad. I think there cannot be a better explanation of the immune dysregulation in COVID infection, and I think we have heard beautifully from two experts, Dr. Abel Lawrence and Dr. Vishad. Now we go to another interesting subject, that as both of them have laid the foundation that the immune dysregulation or an hyperimmune response seems to cause injury in COVID patients, whether critical or moderate or mild infection. Therefore, the role of immunosuppressants and immunomodulators in COVID comes. Also, there are controversies related to that. Does immunosuppression make a patient more susceptible for COVID. So, what are the consensus, what are the controversies in the subject of immunosuppressants and immunomodulators in COVID? So, we have now a brilliant clinician cum researcher, Dr. Padmanabha Shenoy from Kochi. He is the director of the uh, care center in Kochi, and Dr. Shenoy is a beautiful speaker. I would like to we would like all of you to listen to him on this subject of immunosuppressants or immunomodulators in COVID, consensus and controversies. Dr. Shenoy, please welcome.
Good evening, Dhanda sir. Good evening, George Chandy sir. Good evening, everyone. It's a pleasure and privilege back to be at least virtually in Believers. And thanks for the opportunity. So, like Dr. Abel spoke most beautifully, and Vishal also added, the immune system is the key. And modulating the immune system is the only way we can reduce the mortality in one of the major ways we can reduce the mortality in COVID-19. And in March and April, we didn't know that. And by the time we started using steroids and other immunomodulators, the mortality has remarkably come down across the globe. Case fatality from Europe and US, has, as we have seen, from 7 to 8%, it has come down to 1 to 2% now. So definitely immunomodulation is the way forward. So if you look at the COVID stages, as Vishal told, and Dr. Abel also spoke the same thing. Once the virus enters, virus will multiply, and then the viremia will come down, and the host inflammatory response will build up. And when the host inflammatory response build up, that phase is called hyperinflammatory phase. When cytokine release syndrome happens, and the severe immune response destroys the organs. That has been extensively told. So where does the immunomodulation has to play a role is in the third phase of the COVID infection. So first and foremost, I would like to talk about three molecules: hydroxychloroquine, tocilizumab, and steroids. So first and foremost is the most hyped molecule, hydroxychloroquine. Hydroxychloroquine, as all of you know, is a anti-malarial drug, has been extensively used in rheumatology. When people started studying about hydroxychloroquine, everyone knows it increases the pH of the endosome. By doing so, it inhibits the ACE2 expression. Also, it causes, by ACE2 is required for the virus to enter the cell. So chloroquine, by decreasing ACE2 receptor expression, decreases the cellular entry of the virus. Not only that, as it's a immunomodulator, it inhibits all the inflammatory cytokines. So the advantage of hydroxychloroquine is that it inhibits the viral entry during the viral response phase, and it inhibits the cytokine storm. Appears very appealing in the treatment, whichever you need not differentiate which phase it is. So you can give in the phase 1, stage 2, and stage 3, and also in prophylaxis because it downregulates the ACE2 receptor. This is one of the first studies which opened the Pandora's box of hydroxychloroquine and azithromycin as treatment of COVID-19. This was from French. They included patients who were from PCR documented SARS-CoV-2 patients and excluded the whole lot of them. And from one center, they had the patients, so everyone was treated with hydroxychloroquine and azithromycin, and two other centers had control patients. So they had only very few number of patients. If you look at here, there were only 36 patients who were analyzed, and 16 were controlled, 14 were treated with hydroxychloroquine, and 6 were treated with hydroxychloroquine and azithromycin. As they claimed, from day 3 itself, the p-values were significant. At the end of day 6, in the control arm, only 12.5% were negative, while in the hydroxychloroquine arm, it was 57%, and hydroxychloroquine plus azithromycin arm, it was 100%. So the entire world went into a frenzy. There was a panic buying of hydroxychloroquine. Everybody was taking hydroxychloroquine once a week. ICMR came out with a recommendation telling that hydroxychloroquine will prevent you. All the healthcare professionals has to take it. So the charts look very, very convincing. But if you look at the data when it was published, there were 42 patients who were included. 26 of them were on hydroxychloroquine, and 16 of them were on controls. If you look at the 26 patients of hydroxychloroquine, 3 were transferred to ICU, one died and one left hospital and one stopped to hydroxychloroquine due to nausea. But they analyzed in the 16 controls, all patients completed follow-up. So they analyzed 20 patients versus 16. What happened to these 6 patients who became sick or left hospital? We don't know why they were not analyzed. If their data was included, we can very clearly see in the chloroquine arm there was a death and 3 became sick. So the story is not as rosy as they projected it. Then there were more than 500 clinical trials registered across the world in National Clinical Trials Registry. So this was the first data which came out early June in New England Journal, which talked about randomized trial of hydroxychloroquine in post-exposure prophylaxis. What they do, they did very simply, they took household contacts of COVID-19 patients, and within 4 days, they were treated, half of them were treated with placebo, and half of them were given hydroxychloroquine at quite high dose, 800 milligram once, followed by 600 milligram, then 600 milligram daily for 4 days. So quite stiff dose of that was given. And what happened? There was no difference. As you can see here, in the hydroxychloroquine arm, 11% got infection. In the placebo arm, there was 14.3% infection. The p-value was not significant. But there was a major catch in this study. If you look here, laboratory confirmed cases were only 11 out of the total number. Very few were laboratory confirmed cases. Most of them were diagnosed based on symptomatology. 38 of them were diagnosed based on symptomatology. It goes on to say that this study had so it didn't have impact. People thought maybe if you have PCR positivity, they would have given it more value. Then people started, as we know, chloroquine was used not only for pre-exposure prophylaxis, post-exposure prophylaxis, mild disease, moderate disease, and severe disease. The second study, which was published again in September 2020 in New England Journal, they studied the patients who were relatively sicker. For example, those who were receiving either or no supplemental oxygen or maximum of 4 liters per minute oxygen. So half of them were given standard of care and half of them were given chloroquine and some of them were given azithromycin 2. And then they analyzed it. You can see here, out of 1/3 were given hydroxychloroquine plus azithromycin, 1/3 were given hydroxychloroquine, and 1/3 were given 1/3 were given standard of care. As you can see here, there was no difference in the ordinal scale. Hydroxychloroquine versus azithromycin, hydroxychloroquine was controlled, hydroxychloroquine plus azithromycin versus hydroxychloroquine, there was no difference, even versus placebo. All the, you can see all the odds ratios and hazard ratios are overlapping 1, crossing 1, indicating that there was no significance. So they concluded that those patients who were hospitalized with mild to moderate COVID-19, the use of hydroxychloroquine with or without azithromycin did not improve clinical status at 15 days. Then people thought, okay, they have tried in a late disease. Maybe it is effective in the early disease. That's what people started at the time, and still people are treating with hydroxychloroquine. Again, another study which was came from Recovery Trial Group, which has been from UK. They had a huge number of patients, 1,500 patients on hydroxychloroquine and 3,100 patients on usual care. And as you can see here, they had most of them were sick. They were hospitalized patients. 60% were on oxygen and another 16% were on ventilator. Again, almost similar like the earlier trial. Was there any difference? No. There was no difference. Actually, hydroxychloroquine numerically had higher mortality, 25% mortality, while usual care had 22% mortality. So, but the odds ratio, rate ratio was overlapping 1, so it was not again significant. So, chloroquine definitely did not have a role in hospitalized or sick patients. And look at the mortality, same thing, 27 versus 25, and discharge from hospital, there was no difference. Mechanical ventilation, no difference. So, there was no difference at all. Then people, that as I told in New York and other places, when we called our friends, they used to say, yes, it works in early cases. They vouched for it. This was the Annals of Internal Medicine trial, which was published 2 months back. Half of them were given chloroquine and half of them were given placebo. What was the difference? They treated non-hospitalized adults who were under home quarantine and they had very early disease. And most of them were asymptomatic or mildly symptomatic. As you can see here, what happened? This is shows the symptom severity score was not different between placebo and hydroxychloroquine. There was no difference at all. And but only thing was less than 50% of the patients were PCR positive in this study. So it was a clinical study, but they couldn't find any difference between chloroquine versus placebo. Almost similar study where they studied chloroquine with or without azithromycin, like what the French people studied earlier. And 150 patients were given hydroxychloroquine plus azithromycin, 150 were given chloroquine alone, and 150 were given placebo. And it's from Qatar. As you can see here, there is a predominant Indian population. 35 to 40% of Indian population. I am sure most of them will be non-resident Keralized because it's a Gulf country. And so a lot of Indian population was there in the study. That's why it's is of interest to us. And was there any difference? There was no difference in virological cure. No difference at all. Chloroquine, azithromycin, sorry, and chloroquine and placebo. There was no difference at all. So till now what we have told, we have very clearly shown that there is no difference chloroquine makes in severe disease, those on oxygen, those on ventilator, those on home quarantine, or those who are on mild disease. There is no difference. So still people are giving chloroquine for post-exposure or post-exposure prophylaxis. This data was published November 24th, 3 or 4 days back in New England Journal again, a randomized controlled trial of hydroxychloroquine for prevention of COVID-19. What did they do? They took asymptomatic contacts of patients in Spain, and some of them were given hydroxychloroquine and some of them given no drug at all, just observed. So what is the difference? Clinical and laboratory, including they did PCR for almost everybody who were symptomatic, and if they are not symptomatic, at 28 days it was done. So you can see all the risk ratios are overlapping one. PCR positive, PCR symptomatic, hospital virus, all of them, there was no benefit chloroquine could show over just observation. This was the last nail in the hydroxychloroquine story, and hydroxychloroquine very, very clearly proven that it is not effective in neither the treatment nor in the prophylaxis of COVID-19. The data is extremely clear, and there are so this is the meta-analysis which was again published 2-3 weeks back. The strongest available evidence points towards the inefficacy of chloroquine and hydroxychloroquine prophylaxis or in the treatment of hospitalized COVID-19 patients. And now we also have data that even in mildly symptomatic patients, it doesn't offer any protection. So chloroquine story ends there. It tells us how a small study, people wanted something, something they wanted to catch, and the people started using chloroquine right, left, and center in COVID patients, and did not have any benefit. So then tocilizumab, interleukin-6 blockade. Interleukin-6 blockade, we know this was the first trial which was published in Annals of Rheumatic Disease from Italy. It was called CHIC study. CHIC study is, they compared the regimen which was compared with, what did they do? They took severe COVID-19 patients and they gave intensive glucocorticoids with or without tocilizumab. So it was a strategy. They will treat everybody with steroids, and some of them they will escalate the therapy to tocilizumab. And they compared this data with those historical controls who did not receive this regimen. So they had around 160 patients, out of which in the treated group 100% received steroids and 43% received tocilizumab. So around half of them received tocilizumab, and we can see what is the criteria for escalation of tocilizumab, where lack of clinical improvement or worsening of respiratory status despite treatment of steroids. After 2 days, if the patient is worsening, they will give tocilizumab. So what is the average time they waited? They waited for 4 days. So they will give steroids. For 3 to 4 days, they will analyze whether the patient is improving. If the patient is not improving or worsening, they will give tocilizumab. And another very important thing, this is the time, April, May was the time people realized that there is a lot of thrombosis going on. So people started using low molecular weight heparin and therapeutic anticoagulation in these patients also. Interestingly, you can see here how much they were also carried away with the chloroquine. 77% were on hydroxychloroquine. That was the norm, because not giving chloroquine was like a crime at the time with evidence available. So effect of treatment versus control. You can see here, there was no difference. There was a improvement in the tocilizumab arm, either in both in the univariate analysis and multivariate analysis, 1.79, and also it decreases the hospital mortality by around 70% and mechanical ventilation by around 70%. So tocilizumab with steroids was proven to be extremely effective in the treatment of severe COVID-19. But you should always remember that steroids were given in the background and tocilizumab was added on it. So we don't know whether it's the steroid effect which took some time to act or it was the real tocilizumab effect. So people again did the randomized trial. This is from Johnstone's group, which is called BATE Tocilizumab Trial Investigators, again published in New England Journal. At least if some patient had fever, pulmonary infiltrate, and or the need of supplemental oxygen in order to maintain an oxygen saturation 92%. If they have two of this, either they should have fever, pulmonary infiltrates, or oxygen requirement. If two of the three are there, they were treated with tocilizumab, and there was no difference at 28 days of mechanical ventilation or death or discontinuation of supplemental oxygen. So this study told us very clear, this is a independent investigator-initiated trial that showed that there was not much difference in the COVID-19 mortality, and tocilizumab was not very effective. Then came this Kovacta trial. This is a Roche-sponsored trial. It is still not peer-reviewed and published. So COVID-19 associated pneumonia, the primary endpoint was not met. What was the primary endpoint? Clinical status at 4 weeks. Death was 19.7% in the tocilizumab arm versus 19.4% in the plus standard of care arm. Then another study was done by Roche, which was called Impactor Trial. They have randomized 379 patients. Half of them were given tocilizumab and half of them were given standard of care along with placebo, and primary endpoint was 28 days. But this study, unlike Kovacta trial, Impactor trial showed that their primary endpoint was statistically significant. What was primary endpoint? Those who were died or those who were requiring mechanical ventilation. 19.3% in the placebo arm versus 12.2% in the tocilizumab arm. So there was a 7%, 8% difference, and as you can see from day 8 onwards, there was difference in the curves. But also time to clinical failure was also better in patients who were on tocilizumab. But we should always understand that in this slide I very clearly told you their primary endpoint was patients requiring dying or patients requiring mechanical ventilation. But we are not bothered whether they're ventilated or not. We are bothered whether they are surviving. So you can see here that there was no mortality difference, no significant difference mortality in this study also. So effect of tocilizumab appears to be not great, at least by these three randomized Kovacta and the Johnstone study and the Impactor study. It is not great, but it may be effective, and the timing is extremely important. And then came the meta-analysis, which was published 1 month ago. As you can see here in the forest plot, it is overlapping 1. So there is not much effect, but maybe it has a role if the timing has to do something. Chloroquine definitely doesn't have any role. Now coming, so in all these studies, I have told you 3 or 4 studies because things are not clear. But this molecule, dexamethasone steroid, the cheapest of all the molecules, is working wonders. I'm sure those who have treated COVID-19 will vouch for steroids in the second week. So this is the data came from Recovery Collaborative Group, which was published in New England Journal, where they have 2,100 patients in dexamethasone arm and 4,300 in the usual care arm. And so as you can see here, most of them were relatively sick. So 15% were ventilated, 61% were oxygen alone, and no oxygen was given for 24 patients. So this was hospitalized patient. The second week they gave dexamethasone. And if you look at all participants of 6,400 patients, dexamethasone definitely decreased around mortality by around 20%, 0.83. And you can see the confidence interval 0.75 to 0.93. So it is less than 1, and it decreases mortality around 15 to 20%. But then they did the sub-analysis. If you can see here, those patients requiring mechanical ventilation, their patients are only on oxygen, and theirs were not on oxygen. If you see all the confidence interval, mechanical ventilation patients, they had a benefit of around 36%. And those who were only on oxygen, they had 28% reduction in mortality. Those who were not on oxygen, the confidence interval was clearly overlapping 1. In other words, if the patient is sicker, the benefit was more. And in patients who did not have any oxygen, actually it did not give any benefit. And actually, if you can see here, the mortality numerically was higher in dexamethasone arm when compared to usual care arm. So it is not that everybody has to receive dexamethasone, because dexamethasone is immunosuppressant, they can get higher number of infections. Those patients on oxygen or on mechanical ventilation, dexamethasone is useful. Then came this meta-analysis, which was published 1.5 months back in JAMA, and it clearly showed that whatever the steroid you are giving doesn't matter. In the second week, it is usually very effective, and it decreases the mortality by 20-25%. So what is the take-home message? Immunomodulation definitely has to play a key role to play in severe COVID-19, and timing is extremely important. How to time it? It is debatable. In the first, usually when the viremia comes down and the second half, the cytokine storm happens, that is the time. And there is no definite biomarker to suggest this is the time you have to give dexamethasone, but it is a clinical judgment. Dexamethasone definitely has to play a role. Tocilizumab plus or minus may be the choice. So then I thought I will just, this is our data. We have just analyzed yesterday. We have from CARE, we had 130 of our patients were infected with COVID in our cohort, and of which we had around 114 patients with complete data, and we have done antibody tests for some of them. So we were trying to see what happens to our patients, because all our patients are on immunosuppressants, and this is one of the largest in the country currently. This is a country, Indian Rheumatology Association currently is collecting all the data from all the major centers, and we have collected around 450 patients, out of which 130 is from CARE. And of these 130 patients, 2 died. There were 2 mortalities, and both the mortalities were the initial months, April or May. After that, nobody has died. I think people have started learning how to manage these patients now, so the mortality is much less. And so this was the gender, as autoimmune rheumatic disease affect more females. 73% of them were females, and 30% required hospitalization. And mean dose of hospitalization was 10 days. 10 days is because after 10 days the antigen test is done, and so that's the reason for 10 days of hospitalization. And most of them got it from so most of them had rheumatoid arthritis. 33% had rheumatoid arthritis, 8% had some other stuff, and some other lupus, and another 30% had other autoimmune diseases like vasculitis or scleroderma, and 1/3 of them did not have autoimmune condition, like they had either osteoarthritis or fibromyalgia or something else. So most of them had some kind of comorbidities. 22% had diabetes, 33% had hypertension, and 26% had hypothyroidism, and 13% had obesity. 13% were obese, and 6% had background pulmonary disease. They were on drugs. 77% of them were on biologics. 2% of them were, 2 or 3 patients were on 77% on disease-modifying anti-rheumatoid, 2% on biologics, and 6% of them were on steroids. So as expected, 58% of them had mild disease, and 31% had 31% were totally asymptomatic, and 10% of them had moderate disease requiring hospitalization and treatment with steroids and other stuff. 10%. So it is slightly higher than the general population, but acceptable. And 2% mortality, two people died. So the mortality of around 1.6, 1.5% again is similar to the general population. This is the data, fresh data, which we did today. Very interesting. That's why I thought I'll put it across, because we were discussing that antibody response happens in these patients. We did the antibody titers of these patients. As you can see, there's a nucleocapsid kit by the Roche, and we did Johnson & Johnson spike antibody positivity. Spike antibody, we know, is the one which protects against the reinfection. It was only present in 61% of them. We analyzed 31 out of them, only 61% had spike positivity, and 77% had nucleocapsid positivity. It was done around 8 weeks of their, after the antigen or their RT-PCR is negative, or they have recovered from their illness. So that indicates that this is much less than what we expected in the general population. So we have a control group also, we have not analyzed. This is the food for thought. I thought just throw some ideas that antibody positivity is not as high as we expect in these patients, but we know that antibodies are not must to prevent reinfection. They have cellular immunity, memory cells, etc., and even antibody negativity will not tell you that they are going to get reinfection. We don't know much, but this is the data what we have now. I thought we'll just share with you the data what we analyzed today. Thank you for patient listening.
Thank you, Dr. Shenoy. As always, you spoke beautifully with strong clinical data. We have heard so far on the immune system derangements and how the virus tricks the immune system, infects, and Dr. Vishad, after Dr. Abel Lawrence had explained the various immunological mechanisms which play into the picture, and Dr. Shenoy has explained how the immunomodulators and immunosuppressants have to be judiciously used to prevent mortality in severe patients. Now you have heard from us. Now we are going to listen to now the teacher of all four of us, myself, Abel, Dr. Shenoy, Dr. Vishad. All of us have been taught by Dr. Professor Sita Nayak. Professor Sita Nayak was the Dean of SGPGI Lucknow, and she was the head of immunology there. She is currently, she has been also an advisor to the Medical Council of India, and she is also a research advisor to the Apollo Hospitals, Delhi. Madam will speak on towards COVID vaccine challenges and opportunities. I would request you all to listen carefully. You can keep your dinner plate in front of you with the video off, and I would like to say that the questions will be taken up at the end. So this is a very interesting topic. All of us are looking towards a vaccine. So Madam will speak on the realities related to the vaccine. Welcome, Madam.
Thank you so much, Devashish. Thank you so much for asking me to speak. After all those highly technical immunological talks, and I must say that 10 years after retirement, some of it has started going above my head now. But I will try and talk something about COVID vaccine. Maybe not as technical as Abel would like. I'm not going to talk about the constructs and the various RNA, DNA, deletions and insertions and so on and so forth. But more from, in my 70 years plus and 50 years in science, I have not seen as much scientific activity in 10 months as I probably have seen more scientific activity in 10 months than I had seen in the previous 49 years. It is just absolutely amazing. And it's like watching an infant grow from birth to 1 year, you know, that huge period of development. It's like everything is exploding and you really can't keep pace with what's happening day to day, which has its advantages, but I think it also has disadvantages, because I don't know if people are pausing and thinking and reflecting on the various aspects which really need to be looked at when you are talking about a vaccine. So I'm going to start with some history. So what is a vaccine? A vaccine is a substance that contains a dead or a weakened version of a microbe that helps the immune system to recognize and destroy the living microbe during a future infection, and thus helps protect against the disease caused by that microbe. And our acceptance of a vaccine traditionally always has been that you populate and you infect a population and you don't pre-test them. Public health can only work that way, that you immunize everybody and the disease does not occur in that person, or you have a 95% protection against the disease, so that over a period of time, that disease will be eliminated from the community. Of course, we have not really known what is the length of protection that a vaccine provides at the time of its development. But historically, most of the time, a vaccine has behaved like a natural infection. So smallpox infection gave lifetime protection from smallpox. The vaccine also protected you for your lifetime. That was true for most of these diseases. However, the understanding of a vaccine has been modified over time. We obviously have moved away from the dead, weakened antigen. We now use immunogenic components. There are subunit vaccines. There are protein vaccines. there are RNA, DNA vaccines. So that part of the definition has changed. And the vaccine term has become very loose. It's no longer against just a microbe, because we have vaccines against cancer. Even the T-cell activated T cells, which we use for cancer therapy, is called a cancer vaccine. So I don't know how much of a vaccine it is, because it is a targeted product developed for one person, and in spite of that, it's called a vaccine. And does it recognize and destroy? This endpoint has also been diluted, because traditionally a vaccine we would expect would provide, if I took the chance of getting that injection, I'm taking it on the assumption that I will get protection for a defined period of time. And that endpoint has also been diluted over time, and it has happened mostly with vaccines like, especially respiratory virus vaccines, like the flu vaccine, in which 50% protection was considered better than no protection. So going back a little bit in history, the 18th century, 1770 was the first showing of protection by cowpox, but much of the progress in vaccinology occurred in the 19th century, with the term vaccination being introduced in 1802. And many of the bacterial organisms causing diseases were recognized, and gradually people started developing heat-killed vaccines or antitoxins. And it's interesting to note that Indian vaccine trial for cholera at Hafkin was actually conducted in 1893. Subsequently, there were efforts at attenuation, like for Mycobacterium tuberculosis, and it was introduced as a vaccine in 1920s. But this was the first time that we had something called vaccine resistance. 100 years later, this is an important issue, because in the delivery of this vaccine, the vaccine deniers, the vaccine skeptics, and the vaccine resistors are going to play a role in achieving the wonders of this vaccine. So when BCG was first introduced, there was an outbreak of tuberculosis in a batch of children, which is called the Lübeck disaster. And following which, of course, it was proved that it was due to contamination with live tuberculosis bacillus and not because of the BCG itself. There was a big period of vaccine resistance. The 20th century also saw a big burst in virology and establishment of various vaccines to the various viruses, the first one being polio. But the first vaccine for polio also had an outbreak, because the attenuation was not perfect. There was an outbreak of polio among vaccinated children, and there was a huge protest because of this, and a lot of vaccine resistance in the 1950s. But however, Salk, then Sabin, the injectable polio vaccine was created. The US never accepted the oral polio vaccine. They went ahead with the injected version because of the public would not accept the attenuated polio vaccine, oral polio vaccine. And subsequently, MMR was developed and hepatitis B vaccine was developed. And we had the first real success of vaccination, which the global eradication of smallpox in 1978. Then, of course, in the 80s, we had the advent and expansion of recombinant technology. In 1986, we had the first recombinant vaccine, which was for hepatitis B virus, followed by vaccines for a number of other viruses. But also what happened with the establishment of these very successful vaccinations, because children vaccinated at birth did not ever have the diseases disappeared. And the disappearance of disease also caused not only the society forgot the disease. So there was neither personal memory nor societal memory of ever having seen these diseases. And that also has some impact on what we are seeing today. Because in the 21st century, of course, coming more closer to home, we have had successful vaccines for a number of viruses, H. pneumoniae, rotavirus, HPV, which actually prevents a cancer. The average time for vaccine development has been 10 to 15 years, 12 to 15 years, partly because of all these little misadventures along the way, that people became more and more rigid in the processes and the regulatory environment, so that actually you're giving, unlike a drug, something to a normal, healthy adult. And you're giving it to huge numbers of people. So you cannot afford to have missteps. And therefore, these regulatory things that were put on top of the development caused more and more layers of ethical issues and regulatory issues. So the vaccine development was not fast-tracked. And the only one that took less than 5 years was the mumps vaccine, which was developed in the 1960s. Earlier in this century, of course, we had exposure to other coronaviruses, the COVID-1 and the MERS. However, the vaccines never reached market because these outbreaks were controlled by public health measures. And what is to be remembered is that those public health measures, at that time, the public health community had warned that there would be other outbreaks, and therefore it is necessary to have programs in place to have these vaccines for various, the research going on. However, most of the funding for these researchers was shut off, and therefore they were just sort of, some eccentric scientists here and there continued doing a little bit of work on these diseases. But the fact that we had learned something from these vaccine developments has contributed to the very accelerated effort, the current effort, because a lot of these things came out of the cupboard. There was some published data from the early part of the century on these vaccines and their developments and the vectors and so on and so forth. However, what has happened with the elimination of diseases and disease ignorance accelerated vaccine skepticism, and therefore an increasing number of people, especially among the developed world, stopped vaccinating. And therefore they have no memory, lifetime memory of any vaccinations. And this has increased vaccine skepticism and the reemergence of these diseases. You have outbreaks of measles, mumps, all sorts of things in most of the developed countries, Switzerland, Sweden, United States, because children are no longer vaccinated. We should also remember that in spite of much effort, both in terms of man hours and money, we do not have any successful vaccines for the HIV and for tuberculosis. We have unsatisfactory vaccines for influenza. The adult vaccine is not given to children. The children vaccine is not given to adults. The efficacy rate is variable. Every year we have to produce a different one for flu. We have a questionable vaccine for cholera, typhoid. So the organisms are smart. It's not that every vaccine works the same way. We have this protein, we have isolated it, we can clone it, we can produce it in mass and inject everybody and the disease will disappear. I think we have to remember, and this has been reinforced by the previous talks, that many of these viruses and bacteria, they are smart and they are evolving just like us. And so it's not all that easy, or at least we should have a note of caution before we go out and burst the crackers and say that we have a vaccine. And there is also a specific caution for the respiratory viruses because we've not been able to get a vaccine for the RSV yet. So mucosal vaccines, and that's the same thing for cholera, typhoid. Mucosal infected diseases, the vaccines probably are a little bit more challenging. So and then of course the immune response to vaccination, something as to the disease, also is a challenge because there's variability in the immune response itself with sex, age, pregnancy, pre-existing conditions, which Vishal talked about, like diabetes, obesity, not specifically for COVID, but generally. And except for the few of the recent vaccines, we have not looked for a vaccine which will globally work across all age groups and across all peoples. This conversation is probably happening for the first time. And the caution that we might need different vaccines for different age groups and different situations is not being highlighted, because I feel that much of the conversation is not from scientists. We read science and we discuss the science, but the general conversation is not being driven by scientists or public health experts, but is being driven by other agendas. And therefore, vaccine is here, the vaccine is here is the cry that you hear. But is the vaccine here for everybody? Personally, I am well past 70 and I am not sure I'm going to take these first few vaccines. I will wait, right? Because I'm not sure how they're going to work. The other thing is the need for adjuvants. I'll talk about it a little later when I talk about the specific vaccines. Almost all the vaccines we've had so far have all had adjuvants. The regulatory approval of new adjuvants has been the most painful part of vaccine science. So few new adjuvants have been approved for general use. So if that is a regulatory demand and that is a regulatory constraint, all these new vaccines, some of them will need adjuvants. Will we be using existing adjuvants? Will there be new adjuvants? Will they be approved? Because a vaccine consists not only of the antigen, the immunogenic component, but it also, in many cases, needs the adjuvant. And then, of course, there is the route of administration. Because oral vaccines are fine. When it comes to injected vaccines, most of the time we are giving them to young children or people under captive conditions, like elderly people who go to clinics. But a public health distribution of a vaccine, which is injectable, is a challenge which I do not think people really have given much thought to. We eradicated polio with 2 drops on the tongue, which could be given by anybody. Mostly were given by primary school teachers in peripheral areas and ASHA workers. But if you need to have, in this country, 1.3 billion people who need to get 2 injections, 2.6 billion intramuscular injections from the remotest part to the remotest part of the country. I think that is a challenge of the type which we have really not thought about. So at the moment, we have a number of types of vaccines. The WHO list, which is updated every week, has a huge number of vaccines in clinical trials. We have the inactivated, which is a whole virus inactivated vaccine. We have a live attenuated vaccines, which like the oral polio vaccine. We have some subunit vaccines in which only part of the protein is used in the preparation. Then we have vaccines which are carried on a viral vector, and we had both the naked DNA vaccine and the RNA vaccines. So all the various technologies and possibility inactivated, which goes back to the 19th century, and then live attenuated going back to BCG from the early 20th century, to the subunit vaccine, which was the first hepatitis B vaccine of 1976. But the last three, viral vector, DNA, RNA, we haven't had a single approved vaccine in use by any of these technologies. So we are talking about something which is really, really new in the terms of approvals, in the terms of certification, looking at the protocols, and really looking at, how they are going to be used. And when they are deployed in large numbers, how we are going to look for their side effects and how they will behave and how we are going to do the phase 4. All these are questions which we need to keep in mind when we deploy this vaccine. So there are a large number, as you can see, this is probably a little outdated. There are 36, I think now. Total number of vaccines of every type, live attenuated, inactivated, subunit, virus-like particles, replicating vector, non-replicating vector, DNA, RNA, right? And large number of them use an adjuvant. Do not use an adjuvant. They are claiming that they will not need an adjuvant. Oh, sorry. Okay. And some 40, 42, I think in the last list are in various stages of clinical trials, phase 3, phase 2, phase 1, phase 2, 1/2, and so on. Which is a very, very, very quick and rapid progress for any sphere of science. So as of now, there are 10 candidates which are in phase 3, of which the ones which I've highlighted, the 4 which I've highlighted, are the ones which have made announcements about their successful termination of phase 3. And I think Pfizer and Moderna have both already, or maybe later this week, I think Moderna on Wednesday and Pfizer maybe later next week, will be supplying the protocols to FDA for the emergency use authorization. In fact, there are some photographs on the net about an aircraft taking off with these vaccine vials, and they are even predicting, I saw something in today's press saying that they will be inoculating the first workers on December 11th. So the 4th, which is Sputnik, which is a Russian vaccine, has already been approved by their regulator, but we do not know much more about it because they have not really given a lot of information on this thing in the public domain. But I'd like to say a few words about these vaccines. So the Pfizer and the Moderna vaccines are both RNA-based vaccines. Okay, so the major problem with these vaccines, not major problem, both of them have claimed 90% efficacy. And this was the headline news. But we need to pause and look at the design of these studies. Their randomization was done with vaccine group and placebo group. And the end point was 119 infections. Okay. So they were doing weekly follow-up to see who fell sick with COVID. If people became symptomatic, they were tested, and if they were COVID positive, they were declared a case. And then 90% is based on the fact that of the 109, well, first and foremost, unfortunately, the very poor control of the COVID in the US helped them in reaching these figures much faster than they anticipated, I think. Okay, because who thought that by September, October, they would still have such a surge that they would get 190 cases among their vaccinated group, their study group. But out of that 190, 162 or somebody who got our COVID positive disease were from the placebo group, and some 16 or 18, I don't remember the numbers, were from the vaccinated group. So what does that show? And Pfizer's is a similar study. Their protocol is similar. So actually, is this a vaccine? Not in the traditional understanding of vaccine, because it isn't preventing infection. It is not. It is only decreasing the frequency of infection. Okay. So when you extrapolate that 20,000 volunteers to 200,000, 2 lakhs, 2 millions, 2 billions, large numbers of people are still going to get infected. So there are some caveats in these, hurrahs that are going around, which as scientists, we have to be very careful about. We have to keep our eyes and ears to understand the actual data and what is being done. The University of Oxford vaccine, they did not look for symptoms. They actually tested all their volunteers every week. So they've actually looked for infection and that infection thing. But they have unfortunately fallen into some problems because of their announcements about having given the half dose, and the first dose having given better results than giving the full dose. People are now a little skeptical about how did they manage to give the half dose, and they are busy trying to falling over themselves with all sorts of explanations. But they are preventing infection. We don't know much about the Sputnik vaccine. But the reason why I have highlighted some of these is because these are the 3 vaccines which are going to be produced in India and will be available to us. The University of Oxford vaccine and the Sputnik vaccine. The Sputnik vaccine is undergoing phase 3 trials in India, and you hear frequent announcements from the government that the trial is progressing and recruitment is going on. So whenever that comes through, they have promised the production will be at Dr. Reddy's lab, and a large number of doses will be reserved for India. The third vaccine, which is of promise to India, is the indigenous Bharat Biotech NIV, which is just a viral inactivated vaccine. And this is in some ways good for us, because the Pfizer and the Moderna vaccines, being RNA vaccine, need very, very cold. The Moderna needs -70 and the Pfizer needs -20, cold chain right up to the delivery point, which is a real challenge for countries like India. The University of Oxford is a DNA vaccine, so it doesn't need that cold temperature, and as is the Russian vaccine. And the COVAX, the NIV vaccine, is an inactivated viral vaccine, which again the cold chain is much more easy to manage. So these are probably the ones that will go forward, both from the production point and the availability point within our country. Now, a few questions, few issues about these various types of vaccines. Okay. So there are these challenges that we have to keep in mind. The RNA vaccines. Now, the reason why they don't need an adjuvant is because RNA itself acts as an adjuvant. It acts via which RNA binds to TLR3, 7, 8, and therefore activates the immune system. But there are some problems with these vaccines. I won't say problems, but there are some issues with these vaccines. The issue with the RNA vaccine is that if you modify the RNA to increase stability, then you may decrease immunogenicity. If you retain immunogenicity, you may decrease the stability inside the body, and therefore inside the cell, the lifespan in the cytoplasm may be limited. So there are some issues which need to be resolved. Both these vaccines are nanoparticle encased. Not a lot of information has been provided by the company as to what are those compounds. People are only speculating based upon their other published data on previous efforts at vaccine production. So we still need to know a lot. Both companies are claiming that they are getting better nanoparticles, which will obviate the need for freezing and cold temperature, but that is something which we are still waiting for. The DNA vaccines that way have some advantages because the DNA goes into the nucleus. It is more stable, it is less fragile. But there's a difference between these two vaccines. The Oxford vaccine uses chimpanzee adenoviral vector, which obviously is not infective to humans. It has been modified so that it doesn't cause any disease. And therefore, they assume or they see that it will be have a longer lifespan in the body and it will be more effective. The Russian vaccine, which is also on a DNA, on an adenoviral vector, is on a human adenoviral vector. And therefore, a point that Abel referred to is that and this has been seen, that 30 to 40% of people have pre-existing antibodies to adenoviruses due to previous exposure to adenoviruses. So how that will interfere with this vaccine, its uptake, delivery, life in the body, etc., is also something which one will have to observe as things develop. Then the question of mucosal infections. Most respiratory virus vaccine development has been a challenge. Since it seems, as somebody said, HIV, airborne HIV, since it causes a systemic disease and the virus is everywhere, obviously the antibodies will neutralize viruses, various parts of the body. But whether it will prevent primary infection if you are immunity, how much IgA antibodies will be formed, how will the mucosal immunity will be called into play if you're given intravascular injection. I think these are questions that any basic immunologist will ask and will think about. And I think these are issues that will keep coming up as we go ahead with the vaccine story. And of course, finally, as I've mentioned, that acceptance of the vaccine, you need to have either infection or vaccination for 70% of the people. With an R of 3, they have calculated that you need to have 70% immune population for herd immunity minimum. And if 40–50% of people do not accept the vaccine, then how do you achieve that kind of herd immunity? The other issue which I have not put here is that not only is it vaccine acceptance, but vaccine availability. If half the world is too poor to get these vaccines, because the rich countries have banned a lot of them, and if you do not, see, this is a disease, it is a pandemic. So it doesn't really matter whether you live in the back villages of Bihar or in the jungles of Congo, everybody has to be vaccinated. I mean, that 70% herd immunity has to be global. Otherwise, somebody travels somewhere, gets an infection, this can carry on. And therefore, it's both affordability, the widespread deployment, the acceptability. So it's a strange situation because the countries which can afford it probably will not. People don't want to have it, and countries where people happily take it, maybe they cannot afford it. So I'm not quite sure over the next 12 months how we are going to deal with this whole issue of immunizing large parts of the world. So in conclusion, I would say it's certainly a new disease. So there are new paradigms, and it's a situation where we are learning on the job. Every day there is new information coming out, but we need to be driven by the science and not by anything else. And as the vaccine story unfolds, we need to stay alert for surprises, because we have to remember that you are going to immunize large numbers of healthy, normal people, all age groups, all backgrounds, all ethnicities, colors, creed, and we have to have sound mechanisms in place for follow-ups. We haven't even talked about the problems of getting people back for a second injection. Most of these vaccines need 2 injections, and 3 weeks apart. So the challenges are huge. The promises are great, but the challenges are huge. Thank you.
Thank you, ma'am, for explaining us the prospects and challenges of the vaccines. I think Dr. Abel Lawrence has also very actively put his comments on the chat.
Yeah.
You to wait till the end of the last and then we'll take up the questions.
Yeah.
Dr. Vishnu is the rheumatologist faculty at Believers Mission Medical College Hospital. He's did his DM from. He's talking on another very unique aspect of COVID, which is the pediatric COVID, and it is often you have heard of that many of the children with COVID in some places have been reported to have picture like Kawasaki's disease, COVID-2, and many other fancy names have been attributed to the pediatric COVID. Does pediatric COVID uh, represent an immunological mimic of Kawasaki's disease? This is what Dr. Vishnu is going to talk. So after that, we'll take up the questions. Dr. Vishnu, welcome. Please deliver your talk.
Thank you, sir. Thank you, sir, for that wonderful introduction. And I'm sure that Abel sir and Devashish sir are very important in my life because they were the ones who gave my DM degree. And in front of all these eminent speakers, I'm nothing. And this wonderful opportunity for me to talk on a subject which is shared in front of all the eminent pediatricians all over the world, and in fact the immunologist also. So my topic here is mainly pediatric COVID and the hyperinflammatory syndrome associated with pediatric COVID. And my job was simplified by Abel sir and of course Vishad sir to a greater extent. I don't know really how much I can um, give justice to my presentation today because most of it is already covered. And coming to my objectives, it is mainly to make people aware of the spectrum with which the children who are exposed to COVID presents with, and the pathophysiology, which is already covered. I'll be skipping most of it with a brief touch on it, and evaluation as well as management on a quick note on the hyperinflammatory syndrome and its management. So let us say that the children are the privileged group, because even though nearly more than 60 million people are infected all over the world with 1.4 million deaths, the pediatric infectivity and the severity have been limited to the mild to moderate disease severity in majority. In fact, there is a small percentage of children who have a severe disease during or in the post-infective period. And this is attributed predominantly to the low exposure of COVID infection mainly by state and national governments all over the world. They have limited the travel options given to children. The schools were closed and the parks were closed, and this was one of the boon for the children. And also, the children have less amount of comorbid illnesses and habituations like adults. Especially obesity is more prone in adults than in children, and cardiovascular, renal, and immunological diseases are less prevalent in the childhood group. And of course, habituations like smoking, use of tobacco, etc., has is not seen in children, in fact, at least in our country. And there is an altered respiratory flora, which is with ongoing infection, which might already have altered the innate and immune, other adaptive immune milieu in the receptive place of the COVID infection. Then there is an ongoing immunization program, which doesn't have any solid evidence, but has subtle evidence mainly for MMR and other vaccination programs, which could help in the prevention of severe COVID infections. And like Sir said, the innate immune system is very robust, and it is like a knee-jerk reflex, and it clears off the infection immediately in children, which might be one of the common cause for clearance of the infection early in the phase of the infectivity. And lack of immune senescence, which was aptly covered by Abel, sir. Now, the clinical criteria for diagnosing the disease into mild, moderate, and severe is already defined by different groups. Predominantly, the mild disease shows that there is a uncomplicated upper respiratory tract infection with mild symptoms such as fever, cough, nasal and throat congestion, malaise, headache, and without any evidence of hypoxemia or breathlessness. And the moderate disease have some amount of pneumonia with hypoxemia and tachypnea, and they are treated in an inpatient or an hospitalized center. Severe COVID, again, which is less commonly seen in children, presents with dyspnea, cough, central cyanosis, chest wall indrawing, tachypnea, lethargy, acute respiratory distress syndromes, severe sepsis, shock, and multisystem involvement. And it can also, this multisystem involvement can also occur in the later state where the infection have cleared. Coming to the international data, we have seen that in most of the national and international data, we have seen that the disease is limited mainly to mild and moderate severity, whereas one-third of the patient, one-third to 1/4 of the patient are asymptomatic. Severe disease is limited to 10% to, and critically ill patients are only 4% of the entire population. And fever, cough, and upper respiratory infections are the most important clinical manifestations with which they present, even though other manifestations like GI symptoms and fatigue are also seen. But all these data are very heterogeneous, and we have to take individualized state-wise data. Unfortunately, most of it is not available from in India. And the basic clinical features shows that there is a male preponderance for significant disease, with age more than 6 having severe moderate disease or a severe disease. And in those patients, they will have ground glass opacities as well as bilateral pneumonias, which are the predominant findings. However, 1/4 of them have no radiological features. And coming to the indicators of organ damage, CPK, MB, as well as LDH are the most important inflammatory markers that are found in even in association with an elevated CRP and ESR. Coming to the disease characters per se, the incubation period was limited to 10 days, nearly 10 days, and the onset after onset of symptom to diagnosis was lasting only 3.7 days, a mean of 3.7 days. The time to close contact was also 10.8 days, and the nasopharyngeal swab negativity was within 11.43 days. The duration of the hospital stay was limited to 12.97 days in, and it was much shorter, almost by a week in when compared to adults. And there is less incidence of ICU stay in comparison to the adults. And majority of them had a family clusters, and it is again a boon from the limitation of travel exposure. Now coming to the topic per se of multi-system inflammatory syndrome in children, which are associated with COVID, the definition was evolving. The first instances, it was detected in the UK, where there was a rise in the multi-system inflammatory syndrome in a previously healthy child or a previously asymptomatic child who was detected to have COVID. And it was highlighted. Eventually, there rose an increased number of presentation from other centers also. And this led to various defining bodies like RCPHC, PCH, CDC, and WHO defining the disease per se. And the management protocols are also out even from the American College of Rheumatology recently. Now, when to suspect multisystem inflammatory disease? So it is seen in any child who is having a high grade fever and having a clinical and laboratory manifestation like that of a severe systemic inflammation. It should also be associated with a single or a multi-system involvement or a dysfunction. And there can be a possible link to that of COVID-19. Other possible diagnosis should always be excluded. And even though it is a time of COVID, all these children with high-grade fever, systemic inflammation should not be linked to post-COVID or a COVID-related MSC. So it is a diagnosis of exclusion. And the various defining bodies have said that the main inclusion criteria is the presence of fever, which is seen in 100% of the cases. And they will have one or more organ involvement or dysfunction. And it is striking that the WHO has come with at least presence of two of these criteria to call it as MISC. That is rash, mucocutaneous involvement, presence of shock or hypotension, cardiovascular involvement, coagulopathy, and acute gastrointestinal involvement. And this in the presence of at least one of the inflammatory markers like CRP, ESR, fibrinogen, procalcitonin, D-dimer, ferritin, LDH, raised interleukin 6, neutrophilia, lymphopenia, hypoalbuminemia, etc. And there should be a link to SARS-CoV-2 if possible. And always they have mentioned that all the groups have mentioned the need for ruling out other possible infections, alternative diagnosis like malignancies and rheumatological diseases. Now there are different clinical phenotype to this multisystem inflammatory syndrome in children. It can be either overlapping with an infection or without an infection. And 35% are without an overlapping infection or without a Kawasaki-like presentation, where the presentation will be mainly of a shock, ventricular dysfunction with features of inflammation like C-reactive protein and ferritin, high ferritin, hyperferritinemia. And it is associated with the viral serology to SARS-CoV-2, that is in presence of IgG and RT-PCR negativity. Whereas in presence of an acute COVID infection, it can present with active infection marked by an RT-PCR positivity. Respiratory involvement will be the most prominent one, with other inflammatory features involving other organs. And the mortality rate is is much more higher than the one without acute COVID or the one without Kawasaki-like presentation. And when there is a Kawasaki-like presentation, it is seen mainly in 35% of children who are predominantly younger and have mucocutaneous involvement, and they will be having features like that of an incomplete COVID most often. The less commonly they had shock, cardiovascular, myocardial dysfunction, and 2/3 of them have IgG positivity, and 1/3 have both positivity of IgG serology as well as PCR. Now coming to the clinical features, it is divided into mainly common features like fever and mucocutaneous involvement, which with predominant features involving the lip and the oral mucosa. So invariably, like we can say that the lips won't lie, And other features like skin rashes, conjunctivitis, edema of the hand and feet with exfoliations can also be seen. Lymphadenopathy is also a feature which is shared between MISC and KD. Now, the organ-specific manifestations include myocardial dysfunction, conduction abnormality, coronary artery aneurysms, shock, and respiratory involvement like pneumonia, ARDS, gastrointestinal involvement, hepatitis, neurological involvement, which is, even though less common, it can be seen, and renal involvement. Coagulopathy is a feature which can be seen in severe MISC, and it can the clinical features shows that nearly 60% of the children were requiring pressors to maintain the BP, and 71% were admitted to the intensive care unit. The response, despite this severe presentation, was excellent to immunomodulatory therapy, and the mortality was reduced less than 2% when compared to the 0.09% in COVID infections. It is important to note that even in children, comorbidities like obesity was a contributory factor. Cardiac evaluation was the most emphasized one, and the normal cardiac function can be seen even at admission. But eventually, they will develop ventricular dysfunction, and it's suggestive of ongoing myocardial injury. This can also be something which can correlate with what Abel sir and sir told regarding the presence of or injury to the RAS system. The patients present with features of KD have significant cardiogenic shock in children. And the gastrointestinal symptoms were more profound than the cardiovascular symptoms, which led to even laparotomy. And to surprise, they found mainly mesenteric adenitis, intestinal inflammation, ileitis, ileus, and ascites. Coming to the Indian data, the data to from the Indian population are very limited, especially from that of the Kolkata shows that the median age was 5, and children less than 5 years were 60%, with a male preponderance and the median hospital stay of 10 days. The clinical presentation was again confined mainly to fever, mucocutaneous involvement, and gastrointestinal involvement. The IgG positivity to SARS-CoV was seen in 62% with an RT-PCR positivity in 50%, saying that it can be majority were having a post-COVID multisystem inflammatory syndrome rather than during an acute infection. And we can see that nearly 43% had left ventricular dysfunction, and 31% had coronary artery aneurysms. 18% had pericardial effusion. Inflammatory markers were seen in majority. When compared to the international data, the age in international data is little more. That is nearly a median age of 9 years was seen, with ethnicity preponderance to African, Hispanics, and Caucasians than the Asian and Indian population. And obesity was also highlighted as an comorbid illness, even though the respiratory and other comorbid illness were also seen. And the hospital stay was almost similar to that of the Indian population. And 71% had intensive care admission also. And here also we can see that fever was the major symptom. Gastrointestinal symptoms follows. Mucocutaneous symptoms was the next one. And limited was the manifestations of neurological symptoms, which was seen in this international population. And in our data, from the Calcutta, it was shown that IVIG was the most effective immunotherapy, and some required a combination of IVIG with methylprednisolone, and 6.2% required TNF inhibitors also. The risk factors, as I already mentioned, is the age. That here the age group of little more older children can have a risk factor for post-infectious COVID, whereas during infective phase, hyperinflammation can be seen from 3 months to up to 17 years of age. And next is the viral load. chronic comorbidities, and the time of peaking is very important. Most often in a community, after the peaking of a COVID infection, within 1 month, they see that there is increased incidence of multisystem inflammatory syndrome in children. This was aptly noted from the UK data, which showed that within a month of presentation, they had a first incidence of MESC. The pathophysiology is already explained, like the initial viral clearance was by lead to a proper clearance of the virus in the early phase of the acute infection, lead to the complete resolution of the disease without much of damage. Whereas in a hyperinflammatory state with acute infection as well as in the post-inflammatory state, it will lead to various leads to hyperinflammatory state due to various mechanisms as already described by Vishad sir and our Abel sir. So I'm just skipping it because of the lack of time. And always it is important to notice that in a proper immune clearance where the viral load coincides with the interferons as well as the interleukins and the chemokines, the patient will present with a mild disease. Whereas when there is a disadvantaged or an impaired clearance to the virus and impaired response to the inflammatory milieu, it leads to a severe disease and MIS-C. So the ACR has come up with a recommendation for managing patients and evaluation of these patients with MIS-C. It is important that most of the patients should be looked with a broader differential diagnosis, predominantly to rule out other coexisting infections or non-inflammatory pathology like that of malignancy. And fever was the most important symptom, and mucocutaneous symptoms, cardiovascular symptoms, and neurological symptoms, in addition to gastrointestinal symptoms, are features. So patients with suspected MIS-C have a recent peaking of COVID infection in the community and have features of KD and MIS-C to be overlapping in these patients. And they have recommended that all the patients who are suspected of MIS-C should be evaluated in a tiered fashion, on a two-tier fashion. And if only severe symptom of shock or cardiovascular collapse, they should be evaluated together with the tier 1 and tier 2 immediately. So child coming with fever with an epidemiological link to SARS-CoV with any of these two features like mucocutaneous, gastrointestinal, conjunctival, and neurological symptoms, they should be evaluated for MIS-C. And with milder symptoms, they should be evaluated with care with the first evaluation for laboratory evidence for inflammation, mainly increase CRP, ESR, and look for any of the features which will fit into MIS-C. The features which predominantly fit into MIS-C are that of neutropenia, leukopenia, thrombocytopenia, hyponatremia, hypoalbuminemia, and in addition to elevated CRP. If they have any of these, then we should go for a second tier evaluation with prominent care to the cardiovascular and other pro-inflammatory cytokines with SARS-CoV serology and RT-PCR to associate the link with an acute or a post-infectious phase. And always it is important that electrocardiography and echocardiography should be conducted in patients who are suspected with MIS, because as I already mentioned, the MIS patients may have a normal cardiac functions in the initial phase, but eventually in the later phase will develop cardiac dysfunction, leading to a poor outcome, and it can be easily missed. The EKG should always be conducted to look for conduction abnormalities in addition to BNP, troponin T, procalcitonin, ferritin, PT, APTT, D-dimer, fibrinogen, LDH, and cytokine panels. And as we have already seen, the cardiac involvement here is predominantly of a ventricular dysfunction, and a 20% of patients will have coronary artery aneurysms or dilatations. And the difference predominantly between the MIS-C KD and the KD are that MIS-C KD, the children are more older children, and it is more commonly seen in the blacks and Caucasians and Hispanics than in the Asian population. Whereas in KD, the children are more or less less than 5 years of age and of high Japanese or an Asian population. The GA symptoms are more commonly seen in MIS-C than in KD, even though it is also seen in KD. And the myocardial dysfunction is more common. Only 5 to 10% of KD will have KD-related shock syndrome. And inflammatory milieu will be very high in MIS-C with a low total leukocyte count, lymphopenia, and platelet elevation is less common. Whereas in KD, more of thrombocytosis and leukocytosis can be seen. And even though the estimate of coronary artery aneurysms are lacking because of the lack of evidence, it is seen similar to that of KD shock syndrome. And 25% of them with KD who are untreated can have the coronary artery aneurysms. So to summarize, in severe KD, the children will be younger, whereas in MIS-C, the children will be older, with other features like that of encephalitis, neurological involvement, significant myocardial involvement, associated symptoms of sore throat, and significant gastrointestinal symptoms when compared to that of KD. Now, the treatment of KD, treatment of MIS-C was put forward by the ACR with the goal of stabilization of the patient and prevention of long-term sequelae to MIS-C. And to summarize, this management has been divided into various groups. Even though all the children with severe MIS-C, that is presence of shock, KD overlap, toxic shock-like syndrome, should be admitted to the PICU. And they should be having a proper IV access with broad spectrum IV antibiotics. And it is important to seek help. Cardiology consultation, rheumatology consultation, hematology consultation should be sought even when the patient is under proper control, especially when there is hemodynamic compromise and tachycardia disproportionate to the fever and elevation in troponin, BNP, EKG abnormalities, and shock. And in the presence of acute COVID, it should be treated with more of a antivirals along with immunomodulatory therapy like steroids or IVIG. And in history of a remote infection with KD-like features, IVIG along with aspirin should be considered. And myocarditis, we have to always put the patient on IVIG, and a stronger steroid dose should be given when compared to that of the other two. And the steroid should be pulsed for at least 3 days and gradually tapered. If there is presence of coronary artery aneurysms or dilatations, aspirin as well as anticoagulation should be sought for. And it is very important that a daily laboratory evaluation is mandatory for all the children. And at least in the initial phase, the EKG should be taken frequently. In patients with a pre-existing EKG abnormality, they should be connected to a telemetry to look for worsening of abnormality. And in case of persistent inflammatory syndrome, despite IVIG, you should sort for other immunomodulatory therapy, like as already mentioned by Shane sir, with anakinra, tocilizumab, and infliximab. I have already mentioned about the role of antiplatelets and anticoagulants. The recommendation by ACR is predominantly to give a low dose of aspirin up to 81 milligrams with a 3.3 to 5 milligrams per kg per day in patients with the KD and thrombocytosis. The aspirin should be continued till the coronary artery changes normalizes or the platelet normalizes. And in patients with coronary artery aneurysm with a marked by an Z score 2.5 to 10, low dose aspirin is sufficient. However, in patients with a very high score that is more than 10, anticoagulation should be added. And it is mandatory that anticoagulation and aspirin should be continued in the follow-up phase also with proper follow up with a cardiologist in subsequent visits. And the follow up, you should not stop evaluating the patients with at the time of discharge. Echocardiogram should be done on a timely basis, and cardiac MRI is also mandatory to look for evidence of premature cardiac fibrosis and persistent, which will eventually lead to persistent LV dysfunction. In Kerala, we have our state government putting forward the post-COVID clinics and guidelines for treatment of the same. And they have come up with the management guidelines for MIS-C, especially for management of the coronary artery aneurysms and the cardiac manifestations, where they say that the patient should be frequently evaluated with a echocardiogram, at least at the end of first to second week, at 4 to 6 weeks, and even at the end of 1 year after the evidence of cardiovascular injury. Now, the take-home message is that it is not a new disease in our neighborhood, and it is very important to be vigilant and not miss on miss. And we should not take this disease very casually and say that it is a mild infection or mild disease which can come in children, and always investigate to the point. Always call out for help. Multidisciplinary meetings and decisions should be taken in carrying out the treatment protocols. Immunomodulation should be given adequately as and when needed, and follow-up should be taken care of and shouldn't be taken lightly. Thank you.
Thank you, Dr. Vishnu, for the lovely and very comprehensive and the concluding talk on MIS-C and the pediatric COVID disease. There are a lot of questions lined up here. I will mention one or two. One is from Dr. Girija Mohan, had asked Dr. Vishnu why lungs are less affected in MIS-C.
Madam, lungs can be affected in MIS-C, especially when it is associated with acute COVID infections. However, in the subsequent disease like MIS-C associated with KD and post-infectious MIS-C, it is more of a inflammatory, post-infectious inflammatory disease, which leads to more of a cardiovascular dysfunction and due to vascular injury or other organ involvement due to an hyperinflammatory state.
The main reason also is that the children have less ACE2 expression in their lung, so that may be the reason they have less lung involvement. Now, before I hand over the session to Dr. George Chandy, I would request each of the panelists to give their concluding remarks in 2 or 3 sentences. Dr. Abel Lawrence has given very nice comments in the chat box, and Dr. Shenoy had to go early. He had responded to all the queries in the chat box. So may I request first Dr. Abel, Dr. Vishad, and then Madam Sita Nayak to give their concluding remarks, and then I'll hand over to Dr. George Chandy, sir.
I want to start with something which I forgot to say. Dr. Sitanaik mentioned that you normally expect a vaccine-mediated immune response to be similar to that of a natural infection. And in the case of coronavirus, we know that natural immunity is very poor and short-lived. So can it be better than a natural infection? And one of the thing is that typically we try to develop vaccines only when natural immunity existed. And here's the first time we are trying to do something beyond the nature, what nature can do. The thing is that why is it that COVID leads to poor immune response? So we look at antibody response. There are two types. There are long-lived antibodies are produced by long-lived plasma cells, and there are also short-lived plasma cells and plasmablasts which produce antibody for a short period. This is the first type of antibody which is normally produced. The requirement for production of long-lived antibodies or long-lived plasma cell is that they have to pass through what is called a germinal center in the lymph node. The remarkable thing that has been detected in COVID infection is that in COVID infection, there is a total lack of germinal centers. COVID lymph nodes have no germinal centers. And this has been shown to be a defect in follicular helper T cells. These are T cells which are found in the B cell germinal centers and are required for the proper maturation and that is somatic hypermutation and class switching of B cells, which lead to the establishment of long-lived plasma cells which secrete antibody for a long time. And this follicular helper T cell defect is linked to increased TNF alpha production. So in a vaccine setting, these abnormalities of the germinal center will not happen. So it is possible that a vaccine-mediated immune response may be qualitatively and quantitatively superior to a natural coronavirus infection. So which is why if you want to get an immune response, it's better to actually get a vaccine than expect an immune response from the natural infection. And some of the other things I wanted to say is that there is more to RNA vaccines than the Moderna and BioNTech, Pfizer. You can say they are upstarts. They jumped the gun. There are other companies which have been developing RNA vaccines longer than these companies, and they didn't jump the gun because they believe that these vaccines had to be developed more. Like there is another German company which was considered the front runner for RNA vaccine before BioNTech and Pfizer announced their thing. They got delayed because they believed in optimizing the mRNA for stability with selective modification of the chemical modification mRNA. So their vaccine can be stable at 5 degrees Celsius, which is similar to any live virus or most kind of vaccines, which means it does not require any more cold chain than normal vaccine. So, but the thing is, because of this development, CureVac development has been delayed. So you can expect better, more stable, and there another class of vaccine which we have been hearing for a long is something called self-replicating RNA vaccines, where is besides just an mRNA, this mRNA also encodes for the RNA-dependent RNA polymerase, the viral replicase gene, which leads to multiple copies of this RNA being produced inside the cell, so the immunogenicity is enhanced much further. And regarding there were some questions on myocardial involvement. Regarding one of the things in Vishad and I have said that when COVID attacks infected cells, there is a down regulation of ACE2 leading to unopposed angiotensin activation. In myocardial tissue, ACE2 has a significant protective role. In fact, we all know that doxorubicin, anthracyclines leads to myocardial injury. If you transfect ACE2 gene into myocardium, the myocardium is rescued from anthracycline-induced myocardial. That is even an unrelated thing. ACE2 has a protective role, and ACE2 deficient mice are otherwise normal. But moment you infuse angiotensin 2, they lead to a rapid fulminant myocarditis and myocardial damage. Within 4 weeks, they develop myocardial fibrosis and myocardial dysfunction, which is similar. So in COVID, the delayed myocardial injury that we see is quite similar to the myocardial angiotensin 2 induced myocardial injury seen in ACE2 deficient mice. However, there are no studies, not much talk about potential role of angiotensin receptor blockade in animal models of ACE2 deficient thing. In such scenarios, there is ACE inhibitors, ARBs are useful.
Thank you, Dr. Vishad, Dr. Abel Lawrence, for very detailed and explanation of vaccine-related issues. May I request Dr. Vishad and followed by Madam Sita Naik to give their conclude.
Actually, what I think listening to Sita Madam's talk went into a little philosophical mode. What I really feel is that this COVID in many ways have been a rude awakening for all of us. It taught us exactly how much we knew about our body, our immune system, not just the science, but even the social science, how people respond to various social stresses. And it has actually dissected our, not only the biology, but also the society, the very fabric of our society. And I think we are all, we have all improved by learning from this experience. And it is important that we, and as Madam said, there has been so much invested in science in this short 1 year span that that has been in any time in the history of world. And it also shows that we need, if we consider the amount of fear this has arisen, if the humanity needs to survive, it is up to us to invest more, to research more, to understand more, and to prioritize science over politics and all other less important aspects in society. So I think I would like to wind up on this philosophical note. Thank you, sir. Thank you for the opportunity.
Yes, ma'am. Ma'am, you have to unmute.
So yeah, Abel, if you read about the individual vaccine, RNA, and so on and so forth, yes, there's huge amount of information. And yes, I'm aware of all that can be done, and in theory and in practice, and a lot of work is going on. And many of these will come to light, and they will come to the clinic, and we will have the trials in due course. And then we'll have this plethora of vaccines. And as Vishad was also saying, there are so many then human issues involved. Which vaccine goes to whom? It's not just affordability, it's accessibility. It's a lot of issues. We are learning the science every day. But even if you stabilize your RNA, finally, when you inject it into somebody, we still don't know how it's going to respond. Believe me that there have been surprises with vaccines in the past. If we think that we can manipulate it all externally and then inject it and expect magic, I think we have to be cautious. It's not experimental in animals, it's not a laboratory. It is the world and human beings, and our fellow human beings. We can't use them as laboratory animals. And we are trying not to do the type of phase 3 trials we ever did. And we are just trying to put them into the market. Yeah, right? So somebody has gone to court against the Oxford vaccine. You must have seen that case report. Somebody from Tamil Nadu who got the vaccine and said he got some neurological complications. And Poonawalla has put up a strong rebuttal that it had nothing to do with the vaccine. I mean, in today's climate of the media and the way things are, I don't know whether to even believe Poonawalla. Nothing, you can't believe anything that comes in the public domain because there's so much of misinformation. So as scientists, I think we have to keep very alert and just believe in the science and keep in mind that, it's our fellow humans whom we are going to be literally using, in a sense, like laboratory guinea pigs. Because we haven't done that kind of groundwork before. Whatever you say, 20,000, 30,000 phase 3 with this kind of is nowhere near the standards we need to give it to a few billion people. So we have to keep our hearts and our heads together and go with this with caution and with alertness. I guess I'm on the older side of the divide. So I think that some of this is not, the young people are very enthusiastic and they jump in very quickly. I think caution is something that should be in everybody's mind.
Thank you, ma'am. Almost keeping what you said in mind, there's an anonymous attendee's question that says that what could be the strategy for administering vaccine excuse me, to the asymptomatic and symptomatic individuals excuse me, who have already exposed to COVID infection.
Well,
COVID-19.
I know.
Sorry. Yes,
there are lots of discussions. I was also involved in our discussion on all these things. Somebody, I think Vishnu talked about, somebody talked about the antibody-mediated enhancement of disease, AED, which has been reported with the previous experimental coronavirus vaccine. So there are fears. There has been discussions whether we should test before we deploy the vaccine. But if you talk to public health people, a vaccine is a vaccine only if you can go out into the community and give to everybody. The moment you introduce pre-testing, then it's no longer a vaccine. You might loosen that definition any amount that you want. But the moment you say I have to screen all the population, see who has has antibody, who is RNA negative, एक तो affordability, it is not feasible. And secondly, it is not practical. The idea of a vaccination is that you go into the community and immunize everybody, right? So I think, yeah, so that is a little bit of a concern, whether I don't think that if we have to really pre-test everybody, then it's going to get deployed universally. It puts such a huge hurdle in deployment. You know, getting people to take 2 doses itself is going to be a challenge. I mean, how many people have worked in the community and tried to get people to take 1 injection, leave alone 2? And then you say, no, हम blood लेंगे, फिर result wait करेंगे, फिर उसके बाद हम आएंगे, फिर first dose देंगे, forget it. It's not going to happen. I mean, that is with my limited exposure with community work. I'm sure.
Thank you, ma'am. Thank you, ma'am, for I think all of us have combined science and art of COVID science, COVIDology today in a beautiful way. And thank you, everyone, all the speakers, attendees, and my great heartfelt thanks for Dr. George Chandy for taking this initiative and giving me the responsibility to coordinate this session. Sir, I'm guilty of postponing, delaying the session by half an hour. So my apologies, and I hand over to you, sir, with my heartfelt thanks.
Debashish, it has been an absolutely fantastic 3.5 hours. I want to thank all of you. Thank you, Debashish, first of all, for having taken up this responsibility. You called it COVIDology. The immunology of COVIDology is just absolutely fascinating. And that's like some of you said, I think this is going to hold the key for the future. So to Professor Sita Naik, to Professor Abel Lawrence, to Dr. Shanoy, to Dr. Vishad Vishwanath, and to our own Vishnu, and to you, Devashish, we are extremely grateful. If you would give us the permission to edit and use each one of your talks separately, we will be extremely grateful, because we want to listen again and again and learn, because there was so much that you put into these 3.5 hours. And I think if we give Professor Lawrence a chance, I'm sure he'll teach us so much more. It has been absolutely fascinating. So thank you so much. If you give us permission, we will edit it so that each one of these will be a separate webinar for all of us to listen and to try and understand and to learn. We are extremely grateful to all of you. I know you set aside a lot of time for us, but it has been amazing. Thank you very, very much.
Thank you so much, sir. And Abel Lawrence is there, sir. He's an encyclopedia Lawrence. You can call him anytime, sir. He's fabulous. He's kept updated every day, sir. I don't think any of us could read anywhere near what he has read. Thank you, sir.
When you say that, Devashish, I can imagine what Dr. Lawrence's knowledge is. You are a walking encyclopedia in CMC.
Oh, sir. Thank you, sir. Thank you, sir. Thank you for your kind words.
So once again, Devashish, thank you so much for having organized this for us, and we are extremely, extremely grateful.
Sir,
thank you.
Thank you to Believers family. Thank you, sir. Thank you, sir.